Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, Saat Kulesi'nin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, Saat Kulesi'nin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, Saat Kulesi'nin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, Saat Kulesi'nin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, Saat Kulesi'nin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, Saat Kulesi'nin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, saat kulesinin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, saat kulesinin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, Saat Kulesi'nin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, Saat Kulesi'nin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, Saat Kulesi'nin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, Saat Kulesi'nin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, saat kulesinin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, saat kulesinin dinamizmi eşliğinde kemer altında
Say something.
Yes. Let's see. Am I on?
Yes.
Okay, sorun yok.
My microphone is on, right? I'm not.
Yes, yes.
No, I'm not.
Your microphone is on.
Bak
And we
Oğuz.
can see you and your presentation, and also we can hear you. Everything seems perfect. Thank you. Okay.
Then
Do you want me also to turn off my camera, or how would this be organized?
However you wish. We can keep your microphone on and camera on. If you don't mind.
I don't mind. I just wanted to comply with your plans.
Okay. We will mute.
Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, saat kulesinin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, Saat Kulesi'nin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, Saat Kulesi'nin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, saat kulesinin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe seninle. Sadece kampüsle sınırlı kalmayan bir öğrencilik hayatı, üniversitenin bulunduğu şehrin olanaklarıyla şekillenir. İzmir, yılın her döneminde capcanlı olan fuarlarıyla, kongreleriyle, sanatsal faaliyetleriyle, her mevsim sımsıcak kalan geceleriyle ve kordon boyunca akan mutlu insanlarıyla parlayan bir yıldız gibi yaşanası bir kenttir. İzmir'de öğrenci olmak bir başkadır. Bostanlı'daki seyir teraslarında gün batımının renkli gösterilerini izleyebilir, vapurda Ege'nin eşsiz atmosferinin tadını çıkarabilir, saat kulesinin dinamizmi eşliğinde kemer altında tarihi yapıların güzelliğine şahit olabilirsin. Ya da Alsancak ve Konak'ta kordonun keyfini çimlerin üzerinde çıkarabilir, kilometrelerce uzanan bisiklet yollarında sağlıklı yaşayabilir, tarihi asansör veya teleferikten İzmir'in tüm güzelliklerini yukarıdan izleyebilirsin. Her yerde yaşarsın ama İzmir'de yaşadığını hissedersin. Sağlık alanında en gelişmiş ülkelerden biri olan Türkiye'de sağlık kariyerine İzmir Tinaztepe Üniversitesi ile yön vermeye hazır mısın? Bölgenin ve İzmir'in ilk ve tek sağlık temalı üniversitesi olan İzmir Tinaztepe Üniversitesi'nde 22 yıllık tecrübesiyle İzmir'in en büyük sağlık grubu olan Tinaztepe Sağlık Grubu'ndan aldığımız güçle 4 sağlık kuruluşunda alanında uzman sağlık profesyonelleriyle birebir çalışarak tecrübelerinden yararlanabilir, doğayla iç içe spor, sosyal kültürel etkinlikler ve sanat çalışmalarıyla Sosyal Yaşam Kampüsü'nde öğrenci olmanın tadını çıkarabilirsin. Teknolojik altyapısıyla güçlü ve kaliteli eğitim almanın ayrıcalığını yaşayarak laboratuvar ve araştırma merkezlerinde bilime katkı sunabilir, enstitümüzde akademik kariyerini lisansüstü eğitimlerimizle taçlandırabilir, şehir merkezine yakın konumuyla toplu taşıma araçlarıyla kolayca ulaşabilir, rahat ve konforlu misafirhanesinde güvenle yaşayabilirsin. İzmir Tinaztepe Üniversitesi mezunu olarak sahip olacağın uluslararası geçerlilikteki diploma ve Tinaztepe Sağlık Grubu'ndan alacağın tecrübeyle kariyerinde fark yaratabilirsin. Şimdi eğitimine İZTÜ ile yön verme zamanı. Gelecek sende. Tinaztepe
Dear Member of Parliament, dear Member of the Executive Board of UEP, dear TÜBA President, dear Coast Guard Asian Regional Commander, dear Chairman of the Board of Trustees, dear Rector, dear Vice Chancellors, dear Secretary General, dear faculty members, dear members of the press, and dear participants, welcome to Galen Science Days. Today we greet the opening of Galen Research Center with the International Galen Science Days event. We have determined the title of this year as New Frontiers in Vaccines and Therapeutics. Professor Özlem Türeci, one of the inventors of the mRNA vaccine, which is a breakthrough in medicine and the most effective weapon against the COVID-19 pandemic, will give the scientific opening speech, also brings together many speakers who are famous for their internationally influential research. They will give detailed information about the domestic COVID-19 vaccine studies of Professor Mayda Gürsel and Professor Hakan Akbulut. Dr. Evren Alıcı from Sweden will lead us with his latest work on NK immune cells derived from stem cells. Professor Fatih Uçkun, Professor Aykut Türen, and Associate Professor Özgür Şahin from USA will present their studies on developing targeted drugs for cancer. Now, we want to make a brief explanation in Turkish to inform the audience about the language of the event and translation process. Değerli katılımcılarımız, bu etkinliğimiz İngilizce yapılacak olup, çevrimiçi platformumuz içerisinde simultane tercüme hizmeti sunulacaktır. Etkinliği Türkçe dinlemek isteyen çevrimiçi katılımcılarımız, dil seçeneklerinden Türkçe dilini işaretlemelidir. Burada bizleriyle bulunan, etkinliği Türkçe dinlemek isteyen katılımcılarımızın ise telefonlarıyla etkinliğe bağlanabileceklerini ve telefonlarına bağlayacakları kendi kulaklıkları ile etkinliği Türkçe dil seçeneğiyle dinleyebileceklerini hatırlatmak isteriz. And now, I would like to invite Professor Öztürk, Galen Research Center Director, to introduce Professor Özlem Türeci, the keynote speaker of the Galen Science Days Symposium. Professor Öztürk.
Dear participants, welcome to the İzmir Tinaztepe University Galen Days Symposium. This morning, we inaugurated Galen Research Center at İZTÜ. This is a very meaningful beginning for us at these pandemic days. Professor Özlem Türeci qualifies herself as a physician, trained scientist, and translational cancer researcher with hands-on clinical experience and interdisciplinary competencies. She strives to solve multifaceted challenges in medicine by translating innovative scientific and technological concepts from bench to clinical practice. This is a very short and modest description of her. Behind, we can see a strong education in medical studies, a PhD in immunology, clinical practice in hematology, oncology, outstanding science, co-foundation of many high-tech companies including biotech, as well as a celebration of the successful mRNA vaccine launching in their modest home in the Turkish state. She is married to Uğur Şahin, and she describes her work always
in partnership with him. Therefore, Uğur Şahin is both a colleague and a lifetime partner for her. In the past 25 years, Özlem Türeci has focused on novel approaches aiming at exploiting the full potential of our immune system to fight cancer and prevent infectious disease with highest precision and with speed, as you have heard in the past. Her research is about understanding the molecular nature of human tumor antigens, development of novel technologies to exploit and redirect adaptive and innate immune effector functions, development of novel classes of highly potent mRNA vaccines against cancer and infectious disease, monoclonal antibody therapies against novel targets, as well as development of truly individualized cancer immunotherapies. Özlem Türeci has been awarded many times with highest honors, including German Sustainability Award. She has recently been elected to EMBO membership. I sincerely thank Dr. Özlem Türeci for accepting to give the first keynote lecture of our first İZTÜ Galen Days, which I hope will become a tradition. This is a great honor for us and a great commencement to high-quality research aimed at İZTÜ Galen Research Center.
Thank you so much, Mehmet Bey.
It's a great pleasure and an honor to presenting our data and our journey, lifelong journey actually, here on this occasion. And I would like to thank the organizers and you, Mehmet Bey, for this invitation. Before I start, let me just check. Can you hear me fine? Can I proceed?
Yes, please.
Okay, seems to be the case. So what I would like to talk about today is our journey and our story in the development of mRNA vaccines. And this story started I'm trying to command. This story started in the 1990s, actually with a dream which I know is shared by many of my fellow cancer immunologists in Turkey, in Germany, and worldwide, namely the dream to potently attack cancer cells without harming healthy cells with novel immunotherapies such as cancer vaccines, for example. And it was already clear back then that developing an ideal therapeutic cancer vaccine would have challenges. On the one hand, there is the potency challenge, meaning that even very small tumors consist of billions of cancer cells. To combat this overwhelming number and make those tumors shrink needs an extraordinarily effective vaccine and adaptive T-cell immunity, which reflects billions of immune cells. Then there is the tumor heterogeneity challenge. The cancer targets that are recognized by the immune system are different for each and every individual patient. So ideally, a technology would be needed that allows to tailor vaccines to each patient's antigen profile, ideally, or at least for a subset of patients. And that, again, would be associated with a manufacturing challenge, and in particular, if we would go towards truly individualized vaccines, where one would need to be very fast with vaccine manufacturing. And when we try to figure out back in those early years which vaccine format to use, we stumbled over mRNA. mRNA is a way to deliver genetic information. mRNA in general is produced from a DNA template by a simple enzymatic process by in vitro transcription. And the mRNA which you get looks actually exact' like cellular processed mature mRNA with untranslated regions, 5' cap and poly-R tail, which embrace the gene encoding part of the mRNA. And you can generate or create nanoparticulate formulations, meaning wrap this mRNA in order, on the one hand, protect it from ubiquitous RNases, and on the other hand, ensure that you deliver it to the right place. And for vaccines, the right place are antigen-presenting cells of the patient, which then would generate the respective protein, present it to the immune system, and train the immune system to raise immune responses. And back at that time, it was clear to us mRNA has a great potential, but it also has a major drawback. And the technological hurdle of mRNA back then, why it was disregarded by many, was that the amount of protein, which is at the end the bioavailable version that was produced from mRNA, was simply too low and not pharmaceutically sufficient. And so it was clear to us that the next years would be about solving this main problem of the mRNA, namely to produce in cells sufficient protein out of it. And that meant really for us many years of improvement of mRNA on different levels. Some of those levels were really technical. For example, one is shown here. As I pointed out, RNA is produced from a plasmid by in vitro translation, and for this you have to linearize the plasmid with restriction enzymes. And normally, as you know, restriction enzymes cut in their recognition site, and this resulted in mRNA, which after its poly-R tail had some sort of cap which came from the restriction enzyme site. And this was obviously associated, as we found out, with low transcription. So one of the first things we did was to establish a different manufacturing process, namely use enzymes which cut adjacent to the recognition site, and thereby have a true poly-R tail, which immediately translated to higher expression. And then there were other levels on which we could optimize. And these levels were about the cap region of the mRNA, the poly-R tail length, the 3' and 5' UTRs, meaning those parts which actually were not translated at all. And what we did was to go through each of these parts individually and find out which designs would, on the one side, ensure high amounts of protein, such as here, over a long duration. And not in any cell, we did this in human dendritic cells, because the design of these elements and what they do in the cell in terms of protein and translation efficiency is cell type specific. And by optimizing each of those elements and also their combination, we ensured, as compared to the original mRNA designs, a high magnitude of expression. And not only of expression, we, for example, also introduced routing signals into the in vitro translated RNA design, which ensured that the protein produced now much efficiently would route to the right places, to those places which are for loading MHC class 1 and MHC class 2 molecules, thereby ensuring a potent T-cell approach, both helper cells and cytotoxic T lymphocytes. And by combining all those in many years of optimization work, we ensured that this main hurdle of mRNA technology, namely low production of protein, could be overcome. In parallel to our endeavors at the other side of the globe, our colleagues at the UPenn, Katalin Carico and Drew Weissman, worked on an alternative method to ensure high protein production. They were primarily interested in using mRNA for protein replacement therapy, not for immunological approaches, meaning vaccines. And it was known already at that time that mRNA has an adjuvant, has an intrinsic adjuvant effect, which means that it is a natural agonist for sensor molecules, in particular on immune cells, such as dendritic cells. And here it is particularly the uridine nucleotides which have this agonistic activity, and they're binding to these sensors, toll-like receptors 3, 7, and 8, induces a type 1 interferon response and, for example, activation and maturation of dendritic cells, which is desirable for immune therapies, but for Kati and Drew was not desirable because they wanted to use the mRNA for a different purpose. And they found out that this activation not only is an immune activation, it also stalls the transcription of the mRNA. And what they therefore did was to get rid of this activity by replacing the uridines by methyl pseudouridine, and thereby increasing the protein translation activity. Another level of improvement on which we worked was the delivery route of mRNA. And here we found out that the conventional way how vaccines, also in cancer immunology, were tested, namely subcutaneous and intradermal injections, were not the optimal ones. What we assessed was injection directly into lymph nodes, because at that time immunologists already knew that the lymph nodes and the dendritic cells therein are the prime places for inducing efficient immune responses. And we in fact showed in mouse experiments that using this intranodal injection results in strong expansion of antigen-specific T cells, not only in that injected lymph node. These T cells also are detectable in blood, they are circulating, and they also end up in the spleen, the largest lymphatic compartment, meaning that they are able to be expanded and to circulate and have a systemic effect. And what we also observed was that in the injected lymph node, but also in other lymph nodes, we observed, as shown here as red dots, the expansion, the local expansion of T lymphocytes against the respective antigen, which was very fast, of a fast onset once the mRNA had been injected into this lymph node. We wanted to understand this better and wanted to assess what exactly happens to the mRNA. And this is naked mRNA, not wrapped in anything, just in buffer and injected. And when we did this, we observed that the majority of the mRNA, which is labeled here, disappears very fast. Not a surprise, there are ubiquitous RNases, but there seems to be a fast process by which certain cells very fast accumulate this mRNA strongly. And we found out that these are the dendritic cells, the resident dendritic cells in the lymph nodes. And further assessment also supported this observation, in particular CD11, B and C dendritic cells, which are known to be excellent antigen presenters and, so to say, trainers of the immune system in the lymph node, were the ones which took up the RNA and also translated it very efficiently and presented it. And this only worked if we would inject and bring the RNA directly into the lymph node, but not if we would just inject close to the lymph node, draining lymph node, subcutaneously. And we also characterized this process and found out that lymph node resident dendritic cells have a natural mechanism, which is called macropinocytosis, which is very fast and which basically accumulates specifically mRNA-like molecules, so that we could biohack this and use this for our delivery ambitions. An interesting observation was that this, wait a moment, I think I have, yeah, here it is. An interesting observation was that just by injecting mRNA into one lymph node, we would get this systemic effect I showed earlier of T cells not only being induced in that lymph node, but expanding to great magnitudes and circulating. And we asked the question if just injecting one single lymph node, does this effect is the path to potent vaccines, which means large magnitudes of T lymphocytes, by ensuring that not only one lymph node, but as many as possible lymphatic compartments of the body get this mRNA message into the resident dendritic cells, express it, and thereby induce an even augmented immune response. So the next task for us was to make this systemic delivery of antigen encoding mRNA into ideally all lymph node, all lymphatic compartments of the body-wide feasible. And there are different ways of trying to do this. You can take nanoparticles, decorate them with specific targeting molecules, and so on. But we wanted something simple because this was also about clinical translation, and clinical translation means that you should simplify as good as possible. So what we found out was that by mixing mRNA with specific lipids, and for this approach here, we used lipids which were already, which had been already tested in the clinical setting, and just by bringing these lipid RNA nanoparticles into a certain size, 200 to 320 nanometers, very well suited for macropinocytosis, and giving them a certain charge, namely near neutral, made them ending up in lymphatic tissues, in this case shown in the spleen. And as you can see, just by changing the lipid RNA ratio, we were shifting the accumulation from a lung into the spleen, also shown here. So this was now interesting for us and a way to get this body-wide expression of mRNA. And what we found out was that this mRNA is taken up by C11 C-positive dendritic cells, so good antigen presenters and trainers of the immune system in the marginal zone. And these cells loaded with mRNA would traffic into the white pulp, which is ideal for inducing T-cell immune responses. And these C11 cells were the only ones which expressed mRNA, the antigen which was encoded. And they not only did this in spleen, as I showed before, but we also reached by this body-wide approach beyond spleen, also individual lymph nodes, even in the mesenteries, and the bone marrow dendritic cells. An additional effect which was desired and which was mediated by the intrinsic adjuvanticity of mRNA was that we got a local but also systemic type 1 interferon response, which was released from plasmacytoid dendritic cells and from macrophages, and which supported dendritic cell maturation and activation of antigen-specific lymphocytes. And this again then translated in mouse experiments into rejection of established tumors in difficult to treat mouse tumor model systems. So that we asked the question, can we use this, what we call RNA lipoplex vaccines, in order to treat human patients as well? And there is, in addition to finding the right vaccine format, another key question, which is has been tackled in cancer immunology for many years, namely the question: what is the right tumor antigen for cancer immune therapy? And there are schools of thought which say that tumor antigens derived from mutations are the ones which are of interest, for example, based on single nucleotide variations, because this is a short epitope sequence, and most importantly, they are devoid of central immune tolerance. Immune responses might be easier to mount. However, the vast majority of mutations is individual. There are not many mutations which, even in a small subset of tumor patients, are shared and can be used. And on the other hand, there are the non-mutated tumor antigens, cancer germline antigens, or placenta carcinoembryonic antigens, which normally in most of the healthy tissues are not expressed, but are switched in without mutation. Actually, in tumors, they are expressed in multiple tumors. There are shared ones of them, and they, however, have the drawback that they might be flawed by central tolerance, so that the hurdle is higher. So we as scientists preferred to tackle this in an agnostic fashion. So we did not choose between one or the other. We initiated clinical testings of both types of antigens in our RNA lipoplex formulations, so that we have clinical trials ongoing with individualized mutation-based vaccines, but also clinical trials with shared non-mutated antigens. And this is what I want to show you on the next slides. An important information might be that we started with these clinical trials in 2012 and in cancer, and by now, or better to say before the pandemic started, we had treated more than 400 patients in clinical trials, many of them worldwide, with our mRNA-based cancer vaccines. So that there was already some manufacturing and some clinical trial experience. And shows you some of that experience, I would like to start with tumor-associated, non-mutated shared antigens, and show some data from one of our clinical trials. This trial here has been conducted in patients with advanced and metastatic melanoma. And what we did here is to choose 4 different shared tumor antigens: NY-ESO-1, tyrosinase, MHA3, and TPTE. Most of them are known and have been tested over the last decades in different cancer vaccine trials with different formats, DNA, peptide, and protein. We chose these 4 antigens because cumulatively they cover more than 95% of patients with melanoma, even though the composition in each patient might be different of those 4 antigens. And what we did, this was a phase 1/2 study, was to test this RNA lipoplex vaccine with the 6 set of antigens in a dose escalation. And we tested after we found the dose, the vaccine alone and in combination with an anti-PD-1, meaning a immune checkpoint inhibitor. And the patients were first treated with 8 weekly vaccinations as induction therapy, and thereafter monthly continued treatment. And as I said, these are patients with large tumor burden, so advanced and metastatic melanoma, so a treatment effect. An important first learning was that, yes, we get exactly the same delivery of the RNA lipoplexes as we have seen in mice, namely into lymphatic tissues. And you can deduce this here from this PET analysis, where only a few hours after intravenous injection, we see a strong activation and proliferation of immune cells in the spleen here as the largest lymphatic organ. What we also can reproduce in patients is the adjuvanticity. So we get the panel of cytokines we want to induce, for example, interferon alpha, and this is dose dependent, depending on the dose of the vaccine. It's short-lived. The cytokines peak to high but not supraphysiological levels within the first 4 to 6 hours, and after 24 hours they are back again. So a pulsatile release, which does not translate much into any clinical adverse event symptoms. And we got T-cell responses, strong ones, CD4s and CD8s, against our vaccine antigens. And this here is just one of the case reports where we also saw some clinical activity. This is a patient with a recurrent melanoma, and this patient was under checkpoint inhibition, which is the standard of care, pembrolizumab. And as you can see, long treatment with pembro, but under treatment, the tumor and one of the nodules shown here was progressing, and we added vaccination and show shrinkage of tumor. So we have cases of objective responses, and this was associated with high T-cell responses, de novo responses, meaning whereas patients at start of treatment did not have those cancer-specific T cells, they would develop them under vaccination, both with regard to CD4 and CD8 cells. And under continuous vaccination, these immune responses against these antigens were stable and even increased. And this translated into objective responses, which were durable in some of those cases. And we not only saw this in this study with melanoma, we have also other studies ongoing, for example, in head and neck cancer with other antigens which are tumor specific. And what we observe in these studies is that we get high levels of T cell activation here in these examples between 2 and 10% of the peripheral CD8 positive cells. So this was now an example for the shared non-mutant tumor antigens. We are also working on mutant tumor antigens, which are the ones which also by checkpoint inhibitors are activated, so that there is already an understanding that they can be very powerful as for an anti-tumor response. But the challenge is that these antigens are unique to individual patients, and you don't find, because mutation is a random process, and you don't find many which are shared by at least a small number of patients. And therefore, we ask the question, why don't we set up a truly individualized approach? And we did this in this first trial, and other trials are ongoing now. What this means is that you take the patient's tumor, you find out what their mutanome, what their mutations, somatic mutations in this cancer are with next generation sequencing. You select some of those mutations, the best ones, and for this we have developed algorithms. You put those mutations as concateners into mRNA, and you immunize with these mutations. And what we observed is that we get strong immune responses against these mutations in several of those per patient, so polyepitopic responses, and that this not only included CD8 cytotoxic T lymphocytes, but also the helper T lymphocytes, which you would want to have. And another interesting and important observation in melanoma patients, these are patients with recurrent melanoma, and these are recurrences or these triangles prior to vaccination. Vaccination reduces the number of recurrences in these recurrent melanoma patients significantly. One important learning with this approach was to optimize the production and manufacturing process such that we were able to produce, to get fast from genetic sequence, namely the mutanome of the patient, to the administrable vaccine. When we started, this was 3 to 5 months, and in the meantime, it's only a couple of weeks. Meaning that we can win the race against the individual tumor of that patient, even though these are hand-manufactured vaccines. And this learnings helped us a lot for the next challenge to come, which we faced in January 2020, namely the development of the COVID-19 vaccine. So, as you know, we have called the COVID-19 vaccine development Project Lightspeed, because from the very beginning, it was very clear to us this has to go fast, because this is a race against a pandemic virus, but it has to comply with regulatory, scientific, and ethical standards. We started in January with 20 different vaccine candidates, which we compared in preclinical studies. We selected 4 of them for phase 1 clinical testing already in April. The clinical trial started. We narrowed this down to the candidate, which would then go into clinical trial, into phase 3 clinical trials, into the pivotal study. The candidate is called BNT162b2. And in November last year, we had the data which showed us that we have a safe and efficacious vaccine. Meanwhile, there have been emergency use conditional but also full approvals in more than 100 countries, and hundreds of millions of people have been vaccinated with the vaccine, as you can see here. And in the meantime, also more vaccines from other innovators available, so that this was overall a good achievement. Let me skip this one. So what exactly is our vaccine, BNT162b2? It is an mRNA which targets the SARS-CoV-2 spike protein in its conformationally stabilized form. It includes many of those improvements we and others have made. We have used for this purpose not the immunogenic uridine mRNA. We had to use the nucleoside modified mRNA without adjuvant effect, because we had to develop an intramuscular vaccine. For the pandemic purpose, you cannot inject intravenously, because this will not happen in hospitals. And therefore, we needed to combine with a new lipid composition, which turned out to be immunogenic and adjuvanticity mediating itself, so that we had to switch to non-immunogenic mRNA. We have shown in mouse models first by immunizing with this that we get strong, high affinity T cells, and also CD4 and CD8 positive T cells of the Th1 biased cytokine profile, the CD1 ones. And we have also showed that these strong complex immune activation patterns of antibody producing cells and T cells are exactly as we need them to be. What we also showed, and this was also a core of what we wanted to achieve, is even though we were injecting into the muscle, as shown here in these mouse models, the expression of the mRNA happens in the draining lymph nodes, so really at those places where we want them to be. We also could show that we get, irrespective of the doses tested in the clinical trial, and these are now human subjects, high levels of IgG and high virus neutralization titers. And we also get strong CD4 and CD8 positive T lymphocyte responses here in human subjects, even at lowest doses of the vaccine. With this data, we decided to go into phase 3 testing in a randomized study in more than 44,000 participants who were treated with either placebo or with BNT162b2, 30 microgram, 2 doses administered 3 weeks apart, 21 days apart. And the endpoint was to measure 7 days after the dose those cases of symptomatic PCR confirmed COVID-19 disease. And what we saw at the interim analysis was very promising. Namely, we got a clear split between the vaccinated and placebo group. And the final analysis then confirmed this, that we had at that first endpoint vaccine with 95% efficacy against symptomatic COVID-19 in the entire population of tested subjects. And even in the older adults, we had 94%. In the meantime, we have 6-month efficacy data, which is already published. And here, the efficacy is against symptomatic COVID is 91%. Against severe COVID-19 disease, we have 97% efficacy. Now, important questions are: what about duration, and what about variants? An important piece of the answer is that we have shown with sera from subjects vaccinated in our trials that in lab experiments they neutralize or pseudo-neutralize sufficiently robust the new variants, including the Delta variant as shown here. We also have shown, and this is just exploratory data, by mapping the T cell responses of CD8 cells. And this is just an example of 3 patients that T cell responses are polyepitopic. And the epitopes, if you look here, are highly conserved across variants. So in addition to the antibody layer, you have an additional layer on the T cell level, which protects from escape. We also observe, as shown here for the SIREN study, that yes, the virus neutralization titers decline over time. And this is also expected with declining IgGs, whereas the T cell responses seem to be more stable, as shown by the SIREN study. Against most of these variants, which are new, we expect that just a 1/3 dose of exactly the same wild-type vaccine will be sufficient due to this conservation of epitopes. And we have actually already shown this in one of our clinical trials, where we have shown that the 1/3 dose boosts neutralizing titers against the wild type, but also substantially against, for example, the Delta and the Beta variant. And the titers are even higher after the 1/3 dose as compared to the 2nd dose. So this is an important thing to know. What also is helpful to assess cross-immunization against new variants is real-world data. And here there are many studies which are also published in the meantime. This one from Qatar, for example, which shows effectiveness of preventing infection and also preventing severe disease against both the Alpha and also the Beta variant of our vaccine in the vaccinated population. And this is another study from the UK, which shows this as well for the Delta variant, where protection effectiveness is still high. It is expected and also shown that, sure, over time, effectiveness will reduce and drop. And for these cases, we need to be prepared with further boosters with our vaccine. What has also been shown is that longer intervals between the 1st and the 2nd vaccination induce even higher protection against new variants. So for those which have not been immunized yet, this could be a good path to go. And this data here further confirms that on observation that longer prime boost intervals, as expected, they are even more efficient against new variants. We cannot exclude that at some point virus might mutate in a way that escape variants, which are resistant against the current vaccine, the wild-type vaccine, will occur, but we are prepared for this, in particular with mRNA vaccines, where I have shown you that for hundreds of patients, we have conducted, starting from a new genetic sequence, in that case of a tumor, of an individual tumor, to administering the vaccine. We were able to set up a process which works within a couple of weeks. We can build on this and can, once a escape variant occurs, also apply this to such a variant vaccine mutation. With this, I would like to wrap up. I hope I could convince you that mRNA as a technology platform has a huge potential for a couple of therapeutic applications I have shown you and beyond. We could not talk about all of them today, and I hope that we will have an occasion to also talk about other applications such as protein replacement, genome engineering, and allergy tolerization at a later point. So I would like to acknowledge all those who have been involved, and this is just the tip of a huge iceberg of joint efforts, and I would also like to thank you for your interest.
We have about 5 minutes left. I don't know if you have time to answer one or two questions from the audience. Did you hear me? Looks like Özlem Türeci is not hearing us. She cannot hear anything. Technical.
I'm very sorry, I can't hear anything.
All right. I don't know if technical staff can help us. I should at least have a chance to thank her.
Excuse me, can you hear me here?
I can hear you here, but I can't hear a medbay or anything which comes from the.
Hi, Özlem Hanım. Thank you a lot for this very nice talk, and everyone was waiting for it. And I was just going to ask whether you could have one or two questions from the audience, or you are in a hurry as usual.
I have 5 minutes. I have actually another talk right after this one, but I'm happy to answer two questions.
Okay, I have just maybe one question. They are asking whether the success of mRNA vaccines against cancer has been delayed because of your prioritization of the COVID-19 vaccine.
Yes, the cancer vaccine clinical trials have been slowed down, but not really because we have prioritized, but because this was the effect of the pandemic. The lockdowns, patients could not travel anymore, we could not initiate sites, so we found a better way to use those resources, which we had to slow down.
And I have one personal question to finish. When you started the trials, you also had an RNA vaccine that encodes the RBD domain of SARS virus. Did you discontinue this research?
Yes, so we used both and developed them in phase 1 together. And the reason why we had the RB domain was because no one really knew and everyone was scared that we might see an antibody-enhanced disease worsening for this virus. And a way to mitigate that is to have a very small, precise antigen. But in the process, we and others learned from all ongoing trials that this was not really an issue, and therefore we preferred to go with a full spike in order to be better prepared for variants, which was then the higher risk. That's the reason.
Thank you very much. Maybe if you want just to say a few words for saying bye-bye, and thank you again.
Thank you very much. It was a pleasure to be invited and
have a very nice meeting today.
Thank you very much. Bye-bye.
Okay, we would like to go on after this very exciting talk from Özlem Türeci. The next session is about the vaccine candidates that have been developed in Turkey. And I would like to invite İsmail Hakkı Ulus from Acıbadem University to chair the next session.
Organizing committee and the Professor İZTÜ inviting me to participate in the Galen Science Days and opening ceremony to Galen Research Center. This problem. Kusura bakmayın, çift gözlü kullanmak zorunda kalıyorum. Yani gözlüğüm, mesafe uyumlu gözlüğümü unutmuşum. O da yaşlık belirtilerinden. In this section, we have two speakers. We will have two speakers. One of them, Professor Mayda Gürsel. She will, Mayda Gürsel will be focused on the vaccines, and second speaker Hakan Akbulut will focus on adenovirus vaccine. I will, before beginning to Mayda Gürsel, properly introduce to Mayda Gürsel shortly. Mayda Gürsel is professor at the Department of Biological Science at the Middle East Technical University. This research focuses on the natural immune system and vaccines. Mayda Gürsel project from Middle East Technical University Biological Science Department in 1987 and completed our master's degree in the same department in 1999. Receiving this PhD degree from University College London, the School of Pharmacy in 1995. It's about 8 years between 1998 and 2006. He worked at the United States Food and Drug Administration Biological Evaluation and Research Center. She worked there as a postdoctoral researcher 2 years, first 2 years, and then experienced science inspecting biovaccine products. Please go ahead, Mayda Gürsel.
Thank you very much. I'd like to
Mehmet Hoca and all the organizers for their invitation. It was a privilege for me to be in the same session
as Özlem Türeci, Professor Türeci, and I'd like to extend my deepest gratitude to them.
Tremendous effort in bringing out such a
a wonderful vaccine in a very short period of time that probably saved billions of lives. So today I'd like to share with you some of our experience with the VLP vaccines. And right from the beginning of the pandemic, TÜBİTAK initiated the vaccine and drugs platform, and we started, we were one of the groups working in the vaccine field, and we initiated the project as a collaboration between İZTÜ, Bilkent, and Hacettepe University with our MSc and PhD students as our team. So just a very brief introduction about the VLP, or virus-like particle vaccines. These vaccines are not a new platform like the mRNA vaccine. They have been in use since 1986 for the Engerix B against hepatitis B virus. But the VLP platform is continuing to be improved. And for example, in 2021, we have the third generation of VLP-based hepatitis B vaccine licensed by Cybevac. What are the advantages of VLP vaccines is, as the name indicates, these are virus-like particles, and the aim here is to, in a way, mimic the morphology of the virus, the antigen distribution of the virus, minus its genetic component. So when you express viral structural proteins in different cell types, these have a propensity for self-assembly, and you have VLPs produced in the host cells. The antigens mimic the antigenic epitopes of the authentic virus, and they are non-infective, non-replicating, and their particulate nature makes them ideal for draining to the lymph nodes where the first immune activation takes place. Because of their particulate nature, dendritic cells very efficiently internalize these VLPs and present them to T lymphocytes. And experience in the field demonstrated that these VLP vaccines generally induce strong cellular and humoral immune responses. So with the beginning of the pandemic, we aimed to produce the VLP-based vaccine. And for that, we concentrated on 4 structural proteins of the SARS-CoV-2 virus, membrane, envelope, and nucleocapsid. I'll talk a little bit about the spike. We cloned them into dual expression vectors, transfected them into suspension hex cells, and upon transfection, when these genes are expressed, VLPs spontaneously assemble within the hex cells, and the cells secrete these particles into the cell culture medium. So just, I would like to talk a little bit about the spike, because we modified the spike used in our VLPs. The spike protein is a trimeric protein composed of an S2 subunit, trimeric form, and 3 S1 subunits on the top. We can think of this as the S2, the bottom part, as a spring which is squeezed, and on top of it, the 3 S1 subunits reside. But the spike protein isn't very stable, and sometimes this spike spontaneously dissociates into the form shown over here. This is the post-fusion conformation, where the S1s completely dissociate, the trimeric S2 remains, and this will play a part in bringing the viral envelope with the host membrane together. But this, even in the absence of ACE2, the prefusion form of spike can convert into the fusion form, which when this happens, you lose valuable epitopes that would be the target of the immune system. So there must be a way to keep the spike in its pre-fusion conformation and prevent this post-fusion conformation to happen. And that all the studies about the spike protein to scientists like Jason McLelland and Izmir Corbett. Izmir Corbett played a huge role in development of the Moderna vaccine. They were working with SARS and MERS in the past, and they realized that if they introduced two proline mutations into this squeezed spring form of the S2 subunit, the spring will not open, and then the spike would be kept in prefusion conformation. So many vaccine companies, including BioNTech, Moderna, Novavax, and Johnson & Johnson, adopted this prefusion 2P stabilized prefusion spike in their vaccine designs. By the end of May last year, Jason McLelland published another report about the spike protein of SARS-CoV-2 specifically, and he suggested that 4 additional protein substitutions at different locations in this S2 subunit of spike produces a super stable conformation. So when we initiated the study and started producing our VLPs, we incorporated initially the wild type VLP in our wild type spike into our VLPs, but then we moved on to 2P stabilized spike and finally 6P stabilized spike. And usually for protein vaccines, one requires immune adjuvants, immune stimulatory adjuvants. And for that, we tested the utility of a widely used vaccine adjuvant, Alumalone, and the adjuvant we have been using, working on several years, the CpG oligonucleotides alone, or a combination of them for our vaccine formulation. So what we observed when we transfected the plasmids into our HEK cells, you can observe the TEM images over here. The HEK cells started producing the VLPs and just secreted them out of the cell into the culture medium. And as you can see, these look remarkably similar to the authentic virus. We developed techniques to purify the VLPs, including multimodal chromatography, coupled with tangential flow filtration. And when we analyze the VLPs using scanning electron microscopy and atomic force microscopy, these retained their morphologies, and their sizes were similar to the authentic virus, around 120 nanometer in diameter. Whether we incorporated the wild type spike, 2P or 6P stabilized spike, didn't affect the size of the VLPs we produced. So when we characterized our VLPs, the purified VLPs, we looked for expression of these 4 proteins in the VLP. And we saw that regardless of whether the spike was of wild type, 2P or 6P origin, all VLPs expressed the membrane, the envelope, the nucleocapsid to similar levels. However, when we looked at the spike, the wild type VLPs expressed little spike, whereas the 2P stabilized yielded increased spike expression in the VLPs, and we got really good results when we used super stable spike conformation. We also compared the conformation of our spike with inactivated SARS-CoV-2 virus. As I've told you before, the spike is metastable, and you see these dissociated forms of the spike in an activated virus. However, when we use the 6P stabilized form, you don't see these low molecular weight dissociated forms. The spike remained intact. We also recombinant human ACE2 and incubated these beads with different concentrations of the fluorescently labeled VLPs. And we saw that specifically VLPs bound to ACE2 coated beads, but not to the IL-1 beta coated beads. We later produced our VLPs together with Nobel Ilaç in their facility. And one important aspect was to test for the stability of these VLPs. And these passed stress tests, extreme stress tests. We looked at the integrity of the VLPs in our final formulation, and they retained their shape, and they were stable throughout several months. We've been able to follow up to now. So our initial mouse experiments involved 2 different doses of VLPs injected 2 weeks apart with or without adjuvants, either alum alone, CpG alone, or alum-CpG combination. This looks pretty complicated, but the bottom line, what we observed in our studies was that there is one antibody subclass, IgG2A, which is in a way enhanced only if there is one type of T helper cell response. T helper 1 type of cells are required for those, which is desirable for vaccines to protect against viral infections. And IgG2A was supported only in the presence of either CpG alone or CpG alone combination. So from here, we concluded that we should use CpG alum combination based on the antibody findings. And when we compared the wild type 2P and 6P spike expressing VLPs, you can see the results over here. Clearly, the 6P spike expressing VLPs were much more immunogenic than the rest. So we continued our studies with this super stable form. We also isolated the spleen from mice and stimulated them with SARS-CoV-2 specific antigens and checked for 11 cytokines secreted from helper T cells. Again, the summary is over here. Greens represent T helper 2 cells, and we really don't want them in a vaccine because they may support vaccine-enhanced disease. What we want is a Th1 dominated response, shown with the reds over here. And when we use VLPs alone, you can see some Th2, some Th1 response. But when we use CpG plus alum combination, you see that the greens disappear, indicating that no T helper 2 cells are stimulated, whereas really Th1 dominated response is observed. After deciding on the formulation, we continued with our studies, and we determined the effective dose of 50 in mice, and we moved on to different animal models, including rats and ferrets. We immunized them with our VLP vaccine formulation and checked for virus neutralizing antibody response, both in rats and in ferrets. We got evidence that the vaccine induced potent neutralizing antibodies. So around that time, when we were conducting our experiments, the alpha variant, also known as the UK or Kent variant, became dominant in several countries. So we modified our VLP spike protein to accommodate for the alpha variant, and we produced the alpha variant. And surprisingly, use of the alpha variant also increased the spike incorporation in VLPs compared to the wild type. And we carried out some immunogenicity studies with this variant VLP vaccine compared to the wild type. And it was superior in immunogenicity, and also the vaccine generated an antibody response which more broadly recognized other circulating variants in the population. Lastly, we also carried out some protection studies. We immunized a transgenic K12 ACE2 transgenic mice with our vaccine, with 2 different doses, a low and a high dose of vaccine, and then challenged the animals with 3 consecutive days with live SARS-CoV-2 virus. And to summarize the results here, we are seeing the lungs in their half of the lung samples we collected from a healthy mouse, a placebo-treated but SARS-CoV-2 challenged animal, and a low and a high dose vaccine recipient that was also challenged with a live virus. The green indicates the extent of the inflammation in the lungs, and we saw that our vaccine induced a protective effect in K12 ACE2 transgenic mice. So in summary, our study showed that we could produce VLPs. We developed a purification technique that was appropriate for large-scale production. We developed an optimal formulation, and animal results in 3 different animal species showed that this vaccine could induce both neutralizing and binding antibodies to SARS-CoV-2. It supported Th1 dominated immune responses and proved to be protective. Recently, we sent our results, and the journal Allergy accepted this for publication. Based on these results, we initiated the phase 1 clinical trial using 2 different doses of the VLP, original wild-type VLP vaccine, a 3.75 microgram dose and a 4-fold higher dose in humans. Again, the vaccine was formulated with alum and CpG oligonucleotide. 2 doses were introduced 21 days apart. I've a very limited time, so very quickly, I can say that at least in the high dose group, we saw nice antibody responses. These are WHO standards. And according to the standards, the level of antibody, the titers of antibodies in our volunteers was somewhere in between the low and the medium, not as strong as the mRNA vaccine, but there was substantial antibody production. In terms of T-cell responses, from the PBMC of immunized volunteers, we stimulated that or let them alone without peptide, SARS-CoV-2 derived peptide pools, or without the peptide pools, and checked for 12 different cytokines produced from these cells. And what we saw was a nice, mostly Th1 dominated response in the volunteers. No expansion of Th2 cells could be observed, which is good for from a vaccine safety point of view. And our volunteers did not exhibit any severe side effects. And we have now initiated a second phase 2 study by the end of June, which involved 330 subjects and in 3 different arms. One arm is the classical one, spike incorporating VLPs were tested. The second arm it was the alpha variant vaccine alone. And the third arm was the 1 plus alpha combination. And these days we are just evaluating the results of this study for interim analysis. Before I finish, I just would like to show this history of vaccines. Vaccines are the greatest innovations that have impacted global health in a positive way. But because of their success, I think the public got used to it so much that it is taken for granted. And many of these diseases have been forgotten, and people start to think that we can conquer infectious diseases without vaccines by eating, I don't know, pelle pacha and stuff like that, which is not scientific at all. But vaccines are the only way for us to get out of this pandemic. And I just wanted to share with you this slide, which sort of summarizes the situation in the world right now. Most high income and upper middle income countries were lucky enough to be vaccinated, but the vaccination rates in the lower and low income countries are unbelievably low. And unless we vaccinate everyone globally, new variants may arise, and we may have to struggle for several years with this pandemic. So with that, I'd like to thank everyone, but especially TÜBİTAK, Professor Hasan Andal, the head of TÜBİTAK, TÜBİTAK MAM, to Professor Şaban Tekin, to our Science and Technology Ministry, to Nobel İlaç and their teams, to monitor CRO, to the hospitals that are carrying out the vaccination studies and their teams. And I thank all the volunteers that participated in the national vaccine trials, to all of them. But above all, I would like to thank all our young researchers, without whom none of these would have been possible. And I thank you for your
Thank you very much, Professor Yüksel. We have about 5 minutes to ask some questions.
I'm sorry, I don't think
Is there an interest?
Is there an interest to look into multivalent BNPs in the future? Sorry, I'm just reading it from the chat. How is the regulatory pathway for these? Now, because we were at the beginning, we could test at least the immunogenicity of the safety and immunogenicity of the alpha variant in phase 2. But ordinarily, if you look at Moderna, they had to go back to carry out a phase 1 study for the new variant, and they tested the South African variant at the time. So a small safety study is generally required at the beginning. But if you think about the influenza vaccines, in which the vaccine is adopted on a yearly basis, it routinely becomes, in a way, possible to produce the variants without going back to phase 1 once you establish the safety record of different variants. And it may be possible to, especially with a VLP technology, to produce multiple variant spikes in one VLP vaccine, or you can immunize an individual with one type and boost with a different type. That type of approach is also possible. Thank you.
Bir komünikasyon problemi var sanıyorum. İletişim sorunumuz var.
Sorry, we don't hear anything.
Yazmam mı gerekiyor? Ne yapmam gerekiyor? Hangisini? Thank you very much for your nice presentation. We are very happy to listen to you. Thank you very much again.
Thank you. Bye. I wish you the best of Congress. Bye.
MD degree, Tinaztepe University, and trained in the medical oncology at Ankara University. He's currently professor of internal medicine and medical oncology at the Department of Medical Oncology in Ankara University. He has clinical and preclinical research activity in Ankara and is involved in various national and international basic and clinical research projects. He is member of COVID-19 Turkey platform, developing vaccines against SARS-CoV-2 infection. Professor Akbulut has published more than 100 peer-reviewed articles. He is member of several scientific organizations. He is also a UICC fellow since 2008. He serves as a chief of medical oncology department of Ankara University School of Medicine and Cancer Research Institute, Ankara University. Hakan is around.
No, could you start my video? Could you hear me? Okay.
Hakan duyuyoruz.
Okay. Okay, thank you very much, İsmail Hocam.
And before starting my talk, I want to thank the organizing committee and also to thank to Mehmet Öztürk Hocam, and also I want to congratulate Tinaztepe University and Mehmet Hocam for this Galen Research Center. I'm pretty sure that this center will add tremendous work to the scientific basis of Turkey. So let me share my presentation. So I will just, I'm going to briefly overview the adenoviral vaccines against SARS-CoV-2 virus, and also I will give some outlines from our adenovirus vaccine that we developed with help of Tübitag. So the global demand for effective vaccines has triggered a race since the beginning of the COVID-19 pandemic. As a result, a wide range of vaccine platforms has been addressed within 2 years, from the early stage of research through the phase 3 clinical trials and fast-track approvals. We have already approved 4 approved mRNA vaccines in the world. And these vaccines usually include the traditional technologies such as inactivated viruses and protein subunit vaccines, and also the new technologies like VLP as presented by Mayda Gürsel, and also mRNA technologies presented by Özlem Türeci, who are leading with Uğur Şahin and Özlem Türeci, and we are also proud of having these two distinguished researchers in our country. They produce huge amount of mRNA vaccines, and finally the vector vaccines, mainly the adenovirus vectors. And the urgency of the COVID-19 pandemic has resulted in a rush development of viral vector and mRNA vaccines, because it's easy to produce those two vaccines in huge amounts. And soon after the pandemic's beginning, Tübitag has established a platform in March 2020 to develop vaccines and drugs for COVID-19. We, the Ankara University Cancer Research Institute, entered the platform to create an adenoviral vaccine. We are already engaged in developing adenoviral vectors for cancer treatment for more than 20 years, because why we are one of the members of this platform. Adenoviruses are non-enveloped double-stranded DNA viruses having approximately 35 to 40 kilobase of DNA. Those viruses usually cause mild, self-limiting respiratory and eye infections in humans. Almost more than 150 primate adenoviruses have been characterized so far. We have approximately 200 serotypes of adenoviruses. And adenoviruses have been used as gene delivery vehicles since the 1990s. As you see on this slide, adenoviruses have early and late genes, the E1 and E2 and E3, E4, and from L1, L2, L3, and L4, and L5 late genes. The E1 and E3 early genes are especially responsible for the replication of the virus. A typical adenoviral vaccine vector should be replication non-competent. Otherwise, it can be harmful for, especially in people with some degree of immunosuppression. So if you want to use an adenovirus as a vaccine vector, you should make the vector replication non-competent, and you should delete the E1 and E3 genes before moving forward. Therefore, in order to generate an efficient and safe vaccine, adenoviral vector vaccine, the E1 and E3, especially the E1 is the most important one, you should have deleted the E1 genes. And the deletion of those two genes, and so in some platforms, you can also delete E2 and E4 in order to make more room for the large inserts of the antigen genes. And by deleting these two genes, you can have approximately 10 kilobase of room to insert a new gene inside the adenovirus vector genome. And you must insert a strong promoter to drive the vaccine gene. And this promoter is usually a CMV promoter, cytomegalovirus virus promoter in the majority of the vectors. In adenoviral vectors, this is the antigen gene, and also you should include a promoter, a strong promoter like CMV, to produce more, to have more protein expression. And also you should, as I said before, you should have deleted the E1 and E3 genes from the vector to prevent the production of virus inside the cells. And when you design the vector uses the penton and fiber knobs to bind the cell membranes and then enters the cells. So how an adenoviral vaccine works? After injecting the adenoviral vector into muscle tissue or applying intranasal or sublingual, by the way, I should say that the viral vectors have only the chance to use as an apart from the intramuscular systemic use, you can apply as intranasal aerosols, or you can apply as sublingual drops. And you can increase the mucosal immunity, which prevents the settlement of the SARS-CoV-2 in nose and in the mouth. So the viral vectors, the alive viral vectors, are the only vectors that prevents the transmission of the pandemic, the proper virus, and has the chance to stop the pandemics throughout the world. And when you apply the virus and it starts the expression of spike antigen within 24 hours inside the cells, this cell could be a muscle cell or could be an antigen presenting cell. And at the same time, the vector itself triggers innate immune signals. Here, by entering the cells, or the virus itself can also activate the innate response of macrophages and antigen-presenting cells, which makes the induced immune response more strong. And the vector's induction of this innate immune signals further increases the magnitude of immune response against coronavirus. It produces high antibody levels and strong and long-lasting T-cell responses. The sum of the produced antigens are degraded inside the cytosol and presented to the CD8 positive T cells on the cell membrane of the infected cells. And some of the antigens are also secreted directly or indirectly or in apoptotic or necrotic bodies or exosomes and present to the antigen presenting cells. The antigen presenting cells are taking those antigens, migrates to the draining lymph nodes, and where they educate the CD4, CD8, and B cells to produce humoral immunity, antibodies against SARS-CoV-2, and also to produce cytotoxic T cells against SARS-CoV-2 infected cells, or to produce memory CD8 positive T cells for further protection during the pandemics. Adenoviral vectors have some advantages over other vaccines. They cannot integrate into the host genome, so they cannot cause mutations in the cells. They have broad tissue tropism and drive strong expression of the antigen gene. They are highly immunogenic viral vectors. They can induce both B cells and T cells, so they can produce huge amount of antibodies and also huge amounts of T cells. And also they can be produced easily and scalable to global demand. And they are also cheap. And they are highly stable. They can be stored at refrigerator more than for 6 months. However, we have some disadvantages of these vectors. One problem is the pre-existing immunity against the adenoviral vectors, because most of the people in the world have encountered adenoviruses from the childhood. So in some parts of the world, people, up to 50% of people have neutralizing antibodies against adenoviruses. In our country, it's around 20 to 30%, but in some African countries, it's more than 50%, and in China, it's around 50%. And another problem is the vaccine-induced thrombocytopenia, which was reported in March 2021 in people vaccinated with AstraZeneca vaccine. As I said before, there are more than 150 serotypes of adenoviruses in primates. And in humans, there are more than 50 serotypes. In the vaccines that are already in the clinical trials, they used 3 of them. 5 of those vaccines that I will mention briefly after in my following slides, they used human adenovirus serotype 5, and some vaccines used adenovirus serotype 26. By the way, the serotype 5 is the most common serotype of adenovirus in humans. Therefore, the pre-existing immunity against serotype 5 could be an obstacle for the immunity induced by this kind of vectors. And in order to avoid the pre-existing immunity, some people used adenovirus serotype 26, and some others used, like the Oxford vaccine, AstraZeneca vaccine, the non-human adenovirus vaccines, gorilla or chimpanzee adenovirus vectors. As I said, the human adenovirus type 5 is the most used one. However, this serotype is the most abundant in the world. Therefore, there are neutralizing antibodies against these vectors in the majority of people, especially in China, in the Far East, and also in African countries. And the neutralizing antibodies might decrease the vaccine efficacy. In this slide, you can see that the top 10 adenoviral vector vaccines, which are already in the clinical trials, and all the yellow-lined vaccines are already approved for the emergency use and in the world. And they usually use the full-length spike protein of the SARS-CoV-2 virus, and in this the immunity bio companies adenovirus vaccine, they use the fusion gene of spike and nucleocapsid protein together. And some vaccines, they used receptor binding domain of the spike protein. And in 3 of those vaccines, they used perfusion stabilized spike protein, as Dr. Mayda Gürsel mentioned before, because the perfusion stabilized spike protein could be stable after the secretion, and therefore, in order to get efficient humoral immunity against SARS-CoV-2, you must use the perfusion stabilized spike protein. But for the nucleic acid vaccines like plasmid DNA vaccines and adenoviral vector vaccine, you don't need to use perfusion stabilized spike protein. And as you see, the 4 of them already completed the clinical phase 3 trials and are in use in the community. But the new ones, especially they are in the phase 1 level. And if you look at here, they are using the alternative applications, because the approved vaccine vectors are used in intramuscular injections, but here, especially in the United States, there are 3 vaccines used in sublingual or intranasal application, and also 1 chimpanzee adenoviral vector in India, they are using intranasal, and also here another chimpanzee adenoviral vector in the United States, they are applying as intranasal. this vector. And you can see that almost all the vaccines use the same amount of viral vectors. It's around 5 times 10 to 10 to 1 times 10 to 11 viral particles per dose. So the use of this alternating ways of application provides much more mucosal immunity. They are inducing strong secretory immunoglobulin A type antibodies, and which prevents the settlement of coronavirus inside the nose and in upper respiratory tract. So this kind of application has the potential. This kind of application you may know from the polio vaccine, the oral vaccine. You can increase the secretory immunoglobulin A, and you can prevent the transmission of the SARS-CoV-2 from one person to another one. And so I want to give just 1 or 2 slides from this well-known adenoviral vector vaccine. This is the first adenovirus vaccine from the Oxford University and developed with the AstraZeneca. It's a highly effective vaccine, having more than 90% protection from the mild to severe infection. And this paper is from the Chinese vaccine. This is a phase 2 trial. This is why I show this one, because as I said before, in Chinese people, almost 50% of the people having neutralizing antibodies against adenovirus. So if we know that the neutralizing antibodies against adenovirus can decrease the efficacy of the vector vaccine. So, and they just checked this situation. And here, you can see the T-cell responses. We see that there are some decrease in T-cell responses in people having more than 1 to 200 neutralizing antibody levels, but this is still a high response. So it means that even the people, a person has neutralizing antibody against adenovirus, they can still respond to the adenoviral vector vaccine. So it means you may use adenovirus, especially the serotype 5, in developing a vaccine against SARS-CoV-2. And in the Tübingen COVID-19 Turkey platform, we participated with our adenoviral vector vaccine, and we did a different design while preparing our vaccine. We just separated the spike protein. Here you see the spike protein of SARS-CoV-2, which includes 1,273 amino acids, and there are 2 subunits, S1 and S2, and also this is receptor binding domain of the virus, includes almost 200 amino acids. And then we divided this spike protein into 4 subunits, which we named those as A, B, C, and D subunits. You see the protein simulations of the 3-dimensional simulations of these 4 peptides. And here, first we did some bioinformatic analysis before moving forward, and to see the epitope prediction of those peptides to produce antibodies, and also to produce class 1 MHC class 1 immunity T cell responses. And you see here the A subunit, which corresponds to mostly the receptor binding domain of the vector, produces, these are the epitopes producing antibodies. This is the B, this is C, and this is D. The D part mainly corresponds to the S2 subunit of the spike protein. And you see, this is the lower part of, this is the Wuhan original spike protein, and this is our vector design, our subunits of the spike proteins. Here, when we compare the full-length spike protein and our divided subunits, we saw that we can produce much more immunogenic epitopes by dividing the spike protein. And also we checked, can we produce T-cell responses, and you can see here the results, some of those results. This is the A subunit, which binding MHC class 1 binding peptides. And here especially I want to show you this one. When you use the Wuhan original spike protein as a full-length protein, you can produce only 0.28 score for class 1 immunogenicity. Yeah, you can produce only 0.3 T-cell response. But if you separate the spike protein into different parts, you see in our results, the A produces 1.158, the C 1.037, and the B 0.91 T-cell responses. But the D produce nothing in terms of T-cell responses. But as you see the previous slide, the D part can produce immunoglobulins. Here there are 1, 2, 3, 4, 5, 6, 7 epitopes producing antibodies. And as we know that most of the mutations take place in the receptor binding domain of the virus spike protein, which corresponds to our A subunit. So what happens when we inject our vaccine? Just same as the cartoon that I showed at the beginning of my talk. When we inject intramuscularly the vector enters to the muscle cells or into the macrophage or antigen-presenting cells in the tissue. The muscle cells produce spike protein subunits, and those protein subunits taken by the macrophages, and then they are activated, and then they migrate to the draining lymph nodes and then present those antigens to the B cells and or to T cells to induce T cell immunity. And also the macrophages directly uptook the adenoviral vectors. They can process spike proteins and then express those antigens epitopes to the antigen presenting cells, such as it's the same as the other way, and then they can induce both B cells and T cell immunity. So what we had with our preclinical studies, we have shown that our vaccine produced highly significant amount of antibodies against spike protein. And here you see the neutralizing antibody levels of those antibodies. And in this slide, you can see the total IgG against an anti-SARS-CoV-2 IgGs. And after the injection we injected 2 times our vector 2 weeks apart, and then at the 30th day of the vaccination from the beginning, we sacrificed the mice and then took the blood from the heart, and then we observed and increased levels of antibodies. And also we checked the splenic cells in terms of inducing CD8 and CD4 cells, and you can see that the interferon gamma expressing T cells, especially the T helper 1 type of immunity, is increased in the vaccinated group, and also the total CD8 cells with regard to CD4 is increased significantly. But when you looked at the IL-4 positive CD4 T cells, there were no significant difference between the control vaccinated and our adenovirus vaccinated groups, but still we have lower IL-4 positive T cells, which means that with this vaccine we may not have antibody dependent enhancement in people after vaccination. And we had in a significant amount of neutralizing antibodies vaccinated with our vectors. And we did a challenge experiment with AC2 transgenic mice, and here you can see that our vaccine protects the virus infection in almost 80% of the transgenic mice, which is highly significant. And also in terms of pathological examinations, the vaccinated mice have very few infiltration of cells inside the lung tissues. And after finishing our preclinical work, we produced our vaccine in a GMP facility in March 2021, almost 6 months ago. And we are still waiting for the final approval for the clinical trial from the Sağlık Bakanlığı, TİTCK. And hopefully we may to start our phase 1 clinical trial on September 20. Here you see the first adenoviral vaccine vector produced in a GMP facility in this country. This is the first adenoviral vaccine of Turkey, but they are still waiting for the approval from Sağlık Bakanlığı to start the clinical trial. And if we compare our vaccine with others in this same table, in this red line, this is our vaccine. Our vaccine is a mixed vaccine, including 5 vectors with 5 different parts of the spike protein. And we use the same amount of vector per dose. And also we are planning a sublingual application, especially in the phase 2 study, if we can start the clinical trial. And the developer is TÜBİTAK and Ankara University, Turkey. I hope to enter this table within 1 month. And after producing in GMP facility our vector, we continued the new vaccine development studies. We designed new vaccine covering the alpha, beta, gamma, and delta variants of the SARS-CoV-2. And also we continued for modifying the adenovirus itself to decrease the immunity against the adenovirus vectors. So, and our efforts and also the other researchers' efforts make the new adenovirus vectors to evade pre-existing or induce immunity and induce a strong, long-lasting cross-reactive immune response after a single dose, because you have the chance to use a single dose for the pandemic, and also to make more safe vaccines for the global population, especially without the risk of thrombocytopenia and thrombosis, and to make the virus more shelf-stable, and then to keep the virus in the refrigerator maybe more than 1 or 2 years, and to make more cheap and highly scalable. Especially during the pandemics, we need highly scalable vaccine vectors. As the last word, I would say that the COVID-19 pandemic stimulated incredible advancements in vaccine technology and the increased cultural awareness of their importance in public health. Especially this was true at the beginning of the pandemics in Turkey. But nowadays, we cannot see this kind of awareness in our country, unfortunately. And I want to say that in order to prepare the world and Turkey for the next pandemic, the development of highly efficacious and safe vaccines capable of rapid deployment must continue. I want the people from the government and especially from the Department of Health should see this work. Thank you very much.
Hakan, thank you very much. Your nice presentation.
And I want to acknowledge my team in Ankara. Can I share my
slide?
Okay.
Sorry, I forgot to. Because while saying something to the Sağlık Bakanlığı, I forgot to give my acknowledgement. So I want to acknowledge Ankara University Cancer Research Institute, and also I want to thank Pediatric, especially from Professor Hasan Mandal and Professor Şaban Tekin and all these people who helped us too much in developing our vector. And also I want to thank the ODTÜ Bilkent team, Professor Mayda Gürsel and İhsan Gürsel for their kind help, and Professor Osman Erganiş from Selçuk University. And also I want to thank Türkiye Şap Enstitüsü and Türkiye Halk Sağlığı Kurumu, Professor Selçuk Kılıç, who provides us the SARS-CoV-2 mRNA before starting our work. Thank you very much.
Thank you very much. Your nice presentation.
Nice presentation. Also, I'm thankful to sharing your preclinical data related to your vaccine. Good luck for phase 1, phase 2, and phase 3.
Thank you. Hope if we can start, if we get approval from the TCK. We are still waiting. We are waiting for more than 6 months.
Okay. Bureaucracy, as you know, take time.
Yes,
But I think during these pandemic days, it should be accelerated. Good luck again. Thank you very much. Is there any comment or question to Hakan? Again, I think there's no question. Thank you very much again.
Okay, thank you.
Good luck.
I want to say a little word. So I was a volunteer for Mayda Gürsel's vaccine, VLP vaccine. But because of the side effect of VLP vaccine, it's not a real side effect, because the side effect of the Sağlık Bakanlığı, I could not get my vaccine card, and so I cannot get in the airplane, and so I'm talking from the web.
Yeah, this is another side effect.
Yes.
Okay. Good luck again. Thank you very much for your contribution. Sanıyorum aradı.
O yok bende. Yok. Aha gitti. Ne soracağım? Deniyorum şu an. Sen bana verdin şey? Dün derken niye? Bunu vermedin mi şunu? Ben bunu istiyorum. Bana geliyor mu şu an bu? Bakıyorum. Şimdi geldi. Tamam. Başlıyorum.
Participation, either online participants or on-site participants. I'm very pleased to introduce you today's speaker of second session. Our first speaker is Professor Aykut, who is a professor at Georgetown University Lombardi Cancer Center. I know him since 1994, and we were lucky to work together at National Cancer Center with neighbor labs, actually. And he's graduated from İZTÜ University Medical School, and then he continued his postdoctoral research at National Cancer Institute Laboratory of Molecular Biology, Molecular and Cellular Biology. And then he moved to Maryland University in the United States, and he established his team at Maryland University. Then he moved to Georgetown University in Washington, D.C. in 2002. And currently he is a tenured full professor at Georgetown University. He has published several publications in very high impact journals. And he has more than 6,000 citations for them. His research area is focused on cancer research, and he's actually working on the development of small molecules to target specific protein-protein interactions in pediatric sarcomas and prostate cancer. He's also very good teacher and very well-known teacher at Georgetown University, and had several awards for his presentation to medical students. I'm very honored to present him here, and I'd like to thank him for his personal participation to that meeting as well. Stage is yours, Aykut.
Thank you, Neşe. Thank you, Mehmet Hocam. It's a great pleasure to be in this very young university, and hopefully this will be the first of too many to come in the future. I will just jump right into and give you an idea about what we are doing in my lab, what kind of new therapies we're trying to develop. I have to start with this disclosure first. University rules. So the university has several patents. I'm a patent inventor on those patents, and one of those is licensed to a company called Hormone, and I have shares of this company, and they're developing this drug for now in a clinical trial. And I do consulting for a few others too that are not related to today's talk. So this is what we do in my lab. It's just a very broad general outline that is applicable to many, many different disease types. We try to start with some target protein. So whichever the disease that we're going after, we try to select the protein that we can target. And most of the proteins that we are targeting are intrinsically disordered proteins. That's why we couldn't do any crystal structures of those. We can have to do virtual screening. And since they are usually intrinsically disordered and transcription factors or structural proteins, they do not have an enzymatic activity, so we cannot do the high throughput screen either. So our screening comes usually in the form of a direct binding assay. So we screen small molecule libraries for the compounds that will directly bind to our target protein. Once we find those binders, once we know which small molecules are binding to our target protein, then we move on to testing them in biochemical assays, try to see if they bind, but do they also inhibit the function of that protein. If we can come up with inhibitors in a functional assay, then we move them up to cell-based experiments, and we test the function of that specific protein in cell culture. If it works in cell culture as well, then we move them to animal models. We have zebrafish, we have transgenic animals, we have PDX lines and xenografts. And if they all work out, then we end up in a clinical trial. So along this stairway, we start with thousands of molecules in the library, and hopefully at the end we end up with maybe 1 or 2 compounds that are suitable for human trial. And on that path, this is the valley of death that you will hear a lot. What we are doing in the lab needs to be translated to bedside, but there's a huge gap there. Lots of good research, lots of good science gets killed there, and some of them may have potential, but due to many different reasons, they may or may not be translated. So at least one of our studies been to the other side, and some others are down here, they're dying, but we're trying to revive them. So this is a general summary of what we are doing. We focus on pediatric sarcomas. So for the Ewing sarcoma, we've been targeting 3 different proteins. We have a project on EWS-FLI1, I'll tell you about it today. We are targeting GLI and CD99, I'm not going to talk about those. We are targeting a protein called Izmir for osteosarcoma. I will tell you the story on that one. And the rhabdomyosarcoma story is kind of at the beginning stages. I'm going to start with the osteosarcoma story. So there is a protein we selected as a target named Izmir, and it all starts with this. When the experiment was done originally, they put the osteosarcoma cells to the leg of the mouse. It grows and forms a tumor. Then you amputate that leg, get rid of that tumor, and let them live another couple of months. Then you can see the metastasis growing in the lung. They culture those cells out of the lung, put it into a new animal, and go through this cycle 2-3 more times. What you're doing is you're isolating a highly metastatic osteosarcoma cell line. And then when they compare the gene expression between the starting osteosarcoma versus the metastatic one, what you find was at least one of the genes highly expressed there was called Izmir. Izmir is a cytoplasmic protein. It may be in a closed or open configuration. And the classical convention is that when it's open, it binds to plasma membrane, it binds to actin, and this scaffolding is responsible for its metastatic function. At least that's what everybody believes in. So what we did was we made this estrogen as a recombinant protein, and we put him in one of these BioCore instruments. These are surface plasma resonance machines. What you do, you just immobilize your protein in these flow cells. There are different types of them. And then you inject small molecules one at a time. And the machine is so sensitive that it will tell you if a small molecule is binding to your target protein on this microchip. By doing so, we can identify small molecules binding to Izmir. So when we did that, the initial library was about 3,000 compounds. We did identify about 65 compounds just based on binding. So these are the compounds that are sticking to the target protein. And we took those compounds and tested them in different metastasis-related functions. So you're checking the cell motility, we're checking Purex cell invasion, cell viability, zebrafish development. So through those Izmir-dependent functions, we narrowed it down to about a dozen compounds. And then from there, we check the binding kinetics, we check their druggability, and eventually we narrowed down to these 2 compounds that are shown here. And these are our best Izmir inhibitors based on those cell-based assays. Then we move these to animal models. The simplest one we do in the lab for metastasis is called PUMA, pulmonary metastasis assay. In this assay, we inject the GFP, green fluorescent labeled cells, through the tail ring, and then we euthanize the animals and cannulate through the trachea, and we inflate the lungs with this agar solution so that when you take the lungs out, they don't collapse. So you can cut these lungs in small slices and grow them as a small organ culture. And since the cells are GFP labeled, you can monitor their growth over time and test different drugs at different concentrations. So this allowed us to select better working, better inhibiting compounds. We repeat the same experiment by injecting the green fluorescent labeled cells, but instead of euthanizing them and culturing the lungs outside, we did let the animals go, and the treated animals were surviving longer than the untreated animals. So in this experiment, you're putting the cells already into the bloodstream. The cancer cells are given to the animals through IV injection. To mimic the real metastasis a little bit better, we did this experiment in a transgenic animal. So we do have an osteosarcoma transgenic mouse model by deleting P53 and RB in their osteoblast precursors. These animals develop osteosarcoma spontaneously with 100% efficiency. And about 80% of them do develop metastasis to the lungs. So in this experiment, what we did was we let the animals develop the tumor naturally, and then we start the treatment. Then at the end, we monitor the percentage of lung metastasis. The control group was showing about 80% lung metastasis. The drug treatment group was showing about under 40%. So there was about more than 50% benefit in reducing the metastasis. Since this is a transgenic animal, we have no control over where the tumor grows. In these animals, tumors pop on extremities, it sometimes pops on the spine, sometimes in the face. For those strange locations in a mouse, it's not easy to treat them. We actually don't treat them. There's no surgery we can do it. So all these animals have died because of their primary tumor. We were able to evaluate the lungs post-mortem. Since the drug doesn't do anything to the primary tumor, but it is preventing metastasis, we couldn't show the perfect benefit in survival in this model. For that one, we tried a different mouse model. So here what we're doing is we're injecting the tumor to the leg, then the tumor grows, then we do the amputation, take that leg out, and then we start the treatment and see what happens to the lungs. So here in this scale on x-axis, this is time in days. Tumor implantation is day 0. In a couple of weeks, the tumor grows to a sizable, measurable size. Then we do the amputation, then start treating them with the drug for a couple of months, and then we observe them. Untreated group, all the animals have died in 150 days. In the treatment group, we have a good portion survived almost a year. So this was the true experiment where you can take care of the primary tumor by amputation, which is similar to what you would do in a human by doing the amputation, and then treating those patients with the drug to prevent the future metastasis formation. So that was how we developed this compound. We like that. We're trying to push them for clinical trials. But all these small molecule studies, that has also a great benefit in the lab as well, because by running a pull-down and mass spec experiment, the compound gives us clues to how the protein functions, because there's so many unknowns about this protein too. What we do the immunoprecipitation with the DMSO-treated or compound-treated cell lines, and we look at which bands are co-precipitated with estrogen, and which one of those are disappearing. So these are the bands. They normally come down with estrogen, but when you put the compound, they're not coming down anymore. So that is indicating that the compound is preventing this specific protein-protein interaction. So by doing this mass spec analysis, we discovered lots of new proteins that we didn't know they were binding to estrogen. So through that, like these DDX1, DDX3, these are RNA kill cases. Everybody believes that estrogen functions on the plasma membrane, but now what we are seeing is that in addition to this open-close conformation of estrogen working on the membrane, estrogen may bind to mRNA directly, it may be involved in stress granules, it may control protein translation. So all of these functions we didn't know, now that we're discovering it by having this small molecule pointing at us which protein-protein interactions may be happening there. So that's why we're trying to go to the clinic on one hand, but on the other hand, we're going back to the lab and discovering new mechanisms of action for estrogen. Let's go back to Ewing sarcoma. Let me tell you the one that has advanced the most now in clinical trial. Estrogen was the target for osteosarcoma. For Ewing sarcoma, it is again a tumor of the bone and soft tissues. This tumor has a little bit better target. There's a tumor-specific chromosomal translocation. So the EWS gene on chromosome 22 breaks and fuses with FLY1 gene on chromosome 11. So you get this chimeric EWS FLY1. So EWS FLY1 doesn't exist in any of the normal cells. It's only present in the cancer cells. You may have wild-type EWS or you may have full-length FLY1 in a normal cell, but no normal cell has EWS FLY1. So this is your perfect molecular target if you want to go after Ewing sarcoma. The approach was, again, the same. We wanted to go after this EWS FLY because if you get rid of EWS FLY by using antisense RNAs or DNA antisense or siRNA or making a dominant negative protein, one way or another, if you can get rid of EWS FLY or its function, Ewing cells do not grow. Unlike estrogen, which was preventing metastasis, here, loss of EWS FLY results in death of Ewing cells. We did the same screen, came up with this molecule from the Drake binding, and then medicinal chemists started synthesizing lots of derivatives of that lead. So by changing this corner of the molecule, we end up with this compound called YK4279, which was much better than the original lead. That showed really high specificity in EWS FLY expressing cells. The IC50 was less than 1 micromolar. In non-Ewing cells, these cancer cells do not have EWS FLY. The IC50 was always over 20 or 30. When you do a caspase assay, again, you would see the apoptosis development in Ewing cells, but not in non-Ewing cells. Or if you do a reporter assay with an EWS FLY dependent reporter, you see that activity inhibited by using this small molecule. So we have evidence that the small molecule binds to EWS FLY and inhibits its activities in cell-based experiments. And the interesting thing about this molecule is that it has a chiral center. So this molecule will have two different forms. It's like your two hands. They're almost like each other, but they are mirror images of each other. They're not exactly the same. So this molecule has that mirror image, and usually when it's synthesized, you will find them together in a mix. We were able to separate those S and N enantiomers by HPLC. Then when you check the activity, only the S enantiomer was inhibiting EWS FLY activity in the reporter assay or in the IC50, but the R enantiomer was not. So one of these was working, but the other one was not. So it gives you a pretty good negative control in these experiments. It has the same molecular formula, it has the same molecular weight, everything is the same, it's just a mirror image, but it is inactive. So in every experiment we do, we always use that as a negative control to rule out off-target effects. The animal studies was a little bit more complicated. The initial studies were done with xenografts, so it was fine. The mouse with the urine xenografts responded to control cancer, prostate cancer cell lines. They did not. But the PK profile of this drug is poor. In the mouse, you couldn't do that to shrink the tumors. We could only slow down the growth. because it has a very short half-life. So we had to move on to nude rats, where you can kind of like the rats, they put a little jacket, and then we had to give the drug through 24-hour IV infusion that we couldn't do with the mouse, but we could do with the rats. So if you can give a 24-hour IV infusion, in rats you can get some animals shrinking tumor that we couldn't get with the mouse. So with all of that, this was the drug we were able to push for clinical trial, and the company licensed the drug from our university, and they're running the trial. It's going on at 9 different centers in the United States. And this is the publicly available information. I have no connection to the clinical trial. I don't know what's going on as of today, but this was the information that was released last year. The trial was designed like this. This is a new drug. It's not a repurposed drug. This was something that was never given to any human being before. So that's why they start with a very low dose based on animal data, give it to 3 patients. If they do not observe any toxicity, they double the dose and give it to 3 more patients. If there's no toxicity, double the dose, give it to 3 more patients. So they keep increasing the dose until they see a dose-limiting toxicity. In this case, it was neutropenia. At that time, they reduced the dose, tried on 3 more patients, they tolerated well. The first part was just 7-day infusion. So these patients are getting this drug 7 days continuously, 24 hours, because of that poor PK profile. Then you increase the injection to 10 days. They observed that dose-limiting toxicity, lowered the dose a little bit. Now these patients tolerated for 10 days at this dose, and they increased it to 14 days. Now all these patients were tolerating the drug, then they expanded, they added 23 more patients. So at the end of all these patients, the response rate was about 42%. Two patients showed complete remission, all the tumor was gone. 8 patients showed stable disease. And this being a phase 1, all of these are refractory and relapsed viewing patients. So usually those patients do not have more than 2-3 months of life expectancy. So these 2 patients survived. Now they're 18 and 16 months away from their original treatment. So at least those 2 benefited greatly. So now this thing is in phase 2, so we will see how it goes. And in the lab, predicting that those patients, if they get this drug, they may develop a resistance. We're doing the same thing in the lab by exposing the cell lines to the drug at the IC50 level. And then as the cells grow, we increase the drug dose, the cells are growing. The blue line is showing the measured IC50. So as the IC50 goes up, we are increasing the drug dose. So eventually we created a YK4279 resistant cell line in the lab. Now we're studying the pathways that are perhaps responsible for that resistance, so that when the patients get this drug, sooner or later, I'm sure they will develop resistance to it too, so that we can be one step ahead. And whatever we learn from this resistant cell line, perhaps combine those treatments for them in the future when they develop that resistance. So that's sort of 2 summaries that I'm giving you. You know, this story I told you in 5-10 minutes, but this thing all started way back in 2000 with making the recombinant protein. So making the protein was difficult because it was an intrinsically disordered protein. Then we did discover some of the key protein-protein interactions, and we showed that that drug works in prostate cancer, published a couple of papers on that, because I don't think that company would have taken the license if this was a drug for Ewing sarcoma only, because in the US there's only about 300-400 cases a year. So thinking how much it would cost to develop the drug, I'm sure they're looking at the prostate data as well. But eventually, about 20 years later, now we're at the phase 2. And part of the reason is this being a rare tumor, not having too many patients. Part of it is being a rare tumor, not having too much funding, like prostate or breast. But here we are in phase 2, so hopefully we'll get FDA approved soon. So again, this was all done with lots of people over the last 20 years. These are just the current members, and it was funded through Children's Cancer Foundation many years. and we got support from NCI and DoD as well. And if we have time, I can take questions. If not, I'm here all day so we can chat after the seminar. Thank you.
Thank you. It's a really great talk, and we still have time for a few questions. You can ask your questions either from here or our participants who join via online manner, they can also type their questions to the chat box. Are there any questions from audience? Mehmet Hocam?
Thank you very much, Hocam. When you started to select your drug candidates, did you use fully animal model?
So I'm repeating, when you started to screen your candidates in vivo, did you use, you studied this in mouse, did you use mouse tumors or human tumors implanted into mouse?
So when we did the mouse studies, these were all xenografts for Ewing sarcoma because there is no Ewing sarcoma mouse model. But for the osteosarcoma, we did both. We did the xenografts and we did the transgenic. But for Ewing, it was
xenografts of murine tumors.
Xenografts for human tumors. And those for Ewing sarcoma, all the mouse studies and all the rat studies were done in immunocompromised animals. There is no murine Ewing sarcoma.
Okay. Did you see any advantage or disadvantage of this when you went to the human studies?
Well, the doses that we observed in the mouse and rats were somewhat translatable to human studies, so that's how they were decided for the starting dose. And the company who licensed this, when they applied to FDA for an IND and phase 1 trial, FDA requires a large animal study, toxicity study. So I don't know, did they do it in dogs or rabbits, but the company must have done some toxicity study in a larger animal with no tumor.
Do you think if they used primates for that?
In this case, it didn't require, it wasn't required, and
because there's this possibility, as you may guess. So you use a mouse to inject your compound, and the compound will be active against the human tumor, but it may not be toxic because it doesn't recognize normal counterparts or cross-reacting proteins in the normal tissues. So this was my question,
right.
Reason of my question.
So the true target, AWS fly, doesn't exist even in human, in other cells or tissues. It doesn't exist in the mouse either. But just like every other drug, it is very possible that this compound may bind to other proteins and cause side effects. I would expect it will bind to other ETS member transcription factors, FLI1, ERG, or ETB1, ETB4. The bone marrow suppression in the phase 1 probably is due to that. The benefit we observe in prostate is probably due to that. So the compound binding to other ETS members will probably give you that side effect, and I think that is the dose limiting in mouse, and that is dose limiting in rat, and probably that is dose limiting in human as well.
Thank you.
We have a comment from one of our speakers, from Professor Evren Adacı, and he just shared the updated clinical results. Maybe you can summarize, and it's very impressive for sure. Would you like to comment on it?
Well, again, the Ewing sarcoma at this stage, relapsed and refractory, is a really, really difficult thing to treat. So even a patient showing a stable disease is an impressive result. And I'm sure with that, the Children's Oncology Group and FDA will look into this, and they will probably start a new clinical trial with this one as an upfront treatment instead of a phase 1 where everybody failed everything else. Because when the patient comes to that terminal stage, they're very difficult to treat. And so seeing at least some response is very encouraging. So if the drug is tested in an earlier stage, it may show even better response.
Thank you very much. And also I'd like to thank you for your timeline slide. And I wish if our grant supporters see that timeline, because they usually ask from us to have a druggable target and also a drug at the end in 2 or 3 years. So this is really great to see that that timeline was really helpful. Thank you very much again. Our next speaker is Professor Özgür Şahin. Is he connected at the moment?
Yep. I'm there.
Yeah. Hi, Izmir.
Hi.
We are very happy to have you in this meeting. Welcome. And I'd like to introduce you, Dr. Özgür Şahin, who is an associate professor in the University of South Carolina. He completed his PhD in Molecular Biology and Genetics Department as assistant professor, and it was great pleasure for us to meet with him and to see his contribution to science as a young PI. Then he decided to move to United States again, and now he has been working in drug discovery and biomedical sciences department at the University of South Carolina. His main research focus is actually elucidating the mechanisms of drug resistance and metastasis, and he has been working on to develop novel cancer therapeutics actually like our previous speaker. So he has several publications and awards about his research, and we'll be very pleased to listen his recent research. The stage is yours.
Thank you very much. Thank you very much for the kind invitation, also for the introduction. I am really happy to be here today to talk about our recent research. So in the next 25 minutes, I will be talking about our journey from mechanisms of resistance to drug discovery. So I will show how we identify drug-resistant mechanisms, novel targets, and then how we move on discovering novel drug candidates. So here are my disclosures. Only the second one is relevant to today's talk. So our lab is majorly focusing on breast cancer. We have projects on colon and lung cancer as well, but most of the projects currently running in the lab are on breast cancer. It is the most common cancer in women, and there are like more than 500,000 deaths per year, just last year. So it is a heterogeneous disease. So in clinics, we have 3 groups: hormone positive, like estrogen receptor positive, HER2 amplified or triple negative breast cancer, where we have no known drivers, meaning that estrogen receptor, progesterone receptor, or HER2 are not expressed, and we still do not know the driver of this disease. In the beginning of 2000, with the advancement in gene expression profiling, we could classify breast cancer into 5 major subtypes: luminal A, luminal B, ERP2 positive, basal, and normal-like. And the good thing is the clinical classification and the molecular classification are very concordant, meaning that luminal subtypes are hormone positive, P2 positive are HER2 amplified, and most of the basal group is triple negative. So this is a very nice cartoon summarizing actually breast cancer. So what we see here is triple negative, or we can call it basal as well. They are high-grade tumors. They have poor prognosis, and mainstay therapy is chemotherapy. Just in recent years, PD-L1 and antibody-drug conjugate Trodelvy approved. But still, there is a huge problem in this subtype for getting better clinical outcome. And also there is limited therapy options. So today my talk will be on this subtype. And if we look at the luminal A, we see that they are low grade, they have a better prognosis, and endocrine therapy has been the mainstay. And recently we have very good blockbuster drugs like CDK4/6 inhibitors, and recently P110 alpha inhibitors are also approved. And also we have HER2, which is like in between with respect to its prognosis and aggressiveness. Trastuzumab was the blockbuster drug approved 1998, and after trastuzumab, many drugs targeting HER2 have been approved. So we are a Cancer Systems Biology Lab. We are doing an interdisciplinary research. We always start the clinically unmet needs. We start from a clinical question. And I am a molecular and cell biologist by training, and we have been studying drug resistance last 15 years and also metastasis. So we have several cell line models, organoids, and several mouse models representing different drug resistance, like xenografts, syngeneics, transgenics, and also we have patient-derived xenografts. And we use very efficiently genomics, transcriptomics, and proteomics tools, and we are doing a lot of sequencing, reverse-phase protein arrays, and we also do a lot of compound screen, and also we benefit from the proteomic approaches like mass spec. And we do bioinformatics in our lab as well. We utilize patient data sets very efficiently. But whenever we need more detailed analysis, we collaborate with people from bioinformatics field. Then we heavily collaborate with clinicians, medical oncologists, and pathologists. We obtain cancer tissues with follow-up data, and we ask them for several stainings for in situ staining of our target proteins or RNAs. And then we also heavily collaborate with medicinal chemists, because in drug discovery, without medicinal chemists, you cannot go much. So we also do a lot of ADME PK/PD studies with CROs. And then we come back to patients, and that is the final goal, of course. And also after identifying our drug candidates, we also have to find what kind of population, patient population, this drug works. So as you know, in the era of precision medicine, we know very well that not every drug works for every patient. So this is also what we are doing. So what are our current research lines in breast cancer? In TNBC, triple negative breast cancer, we are studying the role of tumor microenvironment in chemoresistance. And we are also developing novel lysyl oxidase inhibitors. Then the second project is both on TNBC and HER2 subtype. We identified and we developed a drug candidate called BO264, which is a TAXI3 inhibitor. And we have licensed this compound to a pharma company. And also we signed a deal with them to develop this drug further, and we are now developing this inhibitor together. And the third project is on hormone-positive breast cancer, which is supported by American Cancer Society. And we are studying the role of phosphodiesterase 4 and our candidate long non-coding RNA in endocrine resistance and metastasis. Due to the time limitations, I will be just talking about this project today. So as I said, TNBC or basal subtype is the most aggressive breast cancer subtype. If you look at the survival of patients in triple negative case, survival is much worse compared to non-triple negative patients. And although TNBC accounts for 15% of the disease, it is responsible for more than 30% of death. So this, I think, clearly shows how aggressive this disease is. And the important thing is TNBC is relatively chemoresponsive in the beginning, up to 45%, but then resistance develops. And very importantly, pathological complete response and good prognosis correlate very well. So how can we use this information? So if you look at this survival curve, if patients are responding, so this is pathological complete response, they have almost perfect survival. And this response is chemo response. So, but if the patients are not responding, if they have residual disease, they have very bad survival. So this means that if we can increase the efficacy of chemotherapy, if we identify the mechanism of resistance and come up with new drugs, we may bring the survival of these patients from here to here. Of course, again, in the precision medicine era, we cannot claim that it will be for all patients. And in the end of the talk, I will tell you how many % of the patients can benefit from the approach we have here. So for this purpose, we modeled the disease in mice. So what we did is we injected the triple negative cells, 231, into mice, and we treated them with either vehicle, meaning the control group, or the doxorubicin. And then they are initially sensitive, like patients, and then we collect sensitive tumors, and we treated them further with doxorubicin, and we obtained resistant tumors. And this is typical waterfall plot showing that the vehicle-treated and resistant tumors have the positive volume fold change, meaning that their tumors are growing, and these are the sensitive ones. And those asterisk ones are sent for RNA-seq, and we identify differentially expressed genes between sensitive and resistant tumors. And we have identified around 500 genes, and we generated doxorubicin resistance gene signature score. And then we asked how relevant this is to the clinics. And we went back to the patients, and we have taken chemotherapy-treated TNBC patients. And when we look at the survival of the patients who have high level of this signature have worse survival. And this has been validated in 3 different data sets. And this clearly shows that our model is very relevant to the clinics. Then if we look at the pathways enriched, integrin signaling was #1, and HIF-1 alpha signaling was also significant. I will come to this later. From the integrins, we had 3 integrins upregulated in resistant tumors at mRNA and protein levels. One of them was integrin alpha-5. And again, if we look at the patients, high level of integrin alpha-5, the patients who have the high level of integrin alpha-5 are doing very bad. So this means they are dying faster. And this is only the case for triple negative or basal subtype under chemotherapy, not without chemotherapy or other therapy, showing that this alteration is specific to this subtype. Again, showing that not every alteration will happen in every breast cancer. So then again, come back to this HIF-1 alpha signaling. As you might know, HIF-1 alpha signaling is major regulator of hypoxia. And we suspected maybe we have huge hypoxia in resistant tumors compared to sensitive. And CA9 is a marker. Again, at mRNA protein level, we have seen a very significant increase of hypoxia. And then again, going back to the patients, again, we see that patients who have doxorubicin resistance gene signature score, we have enrichment of hypoxia. And whether hypoxia is causal, we tested with an assay where we culture 231 cells with normox condition or hypoxic condition, and we have given doxorubicin in a dose-dependent manner. And as you can see, in hypoxic conditions, cells are dying less, meaning that the cells are more resistant to doxorubicin, showing the causal involvement of hypoxia. Then HIF-1 alpha is a major transcription regulator, and there are many targets of HIF-1 alpha, and what we have seen is out of 40 targets, almost 30 of them were upregulated in our resistant tumors, and one of them is lysyl oxidase. This protein took our attention because this has been a very well established regulator of metastasis, but not much known in the context of resistance. So we validated its upregulation in resistant tumors, and in collaboration with Hacettepe University, we stained the patients' tumors, again chemotherapy-treated TNBC patients for LOX, and as you can see, patients having high LOX expression have the worst disease-free survival, again similar to our mouse model. And what is LOX doing? LOX is synthesized in the cell, and it is secreted, and it actually experiences a lot of post-translational modification. The major one here is BMP1 cleavage. When LOX is cleaved, it is active, and it does its function, which is ECM cross-linking, like cross-linking collagen and elastin, and providing tissue stiffness as well as providing signaling to the cells.
And since LOX is upregulated, we thought that maybe resistant tumors are more stiff and there is more fibrillar collagen.
There is more fibrillar collagen. With serious red staining, we show that actually resistant tumors are more stiff. And beginning of 2000, this review article published, and what this paper says is from tumor edge to tumor core, if you move, what you see is oxygen content decreases and stiffness is increased. So this is exactly what we are talking about here. And also, if you look at the patients, again, patients who have high LOX expression, enrichment of hypoxia, and the downstream of integrin signaling population. And then we look if there is a positive correlation between LOX and the integrin alpha-5, which I showed in the beginning, and FN1, which is the ligand of integrin alpha-5. So these are also known HIF-1 alpha targets. And here we analyzed 10 different breast cancer data sets, and these are the Pearson correlation coefficients. So with HIF-1, there is kind of good correlation, but not perfect. But if you look at the correlation between these two and LOX, we have much stronger correlation
LOX might actually regulate the transcription of these genes. And then we tested this experimentally, and this is just a control experiment showing that under hypoxia, LOX activity increases, and then we use LOX inhibitor, it decreases. And then when we induce hypoxia, LOX expression increase, ITGA5, FN1 also increases. And this is also the case at the protein level, and also downstream FAK SARC signaling also increased. And if you look here, we have two different siRNAs to block LOX. And when we block LOX, ITGA5 and FN1 goes down. Also at protein level, also with inducible shRNA. So all these data show that hypoxia-induced LOX actually hyperactivates integrin signaling by regulating integrin alpha-5 and it is ligand FN1, fibronectin. So then we ask if LOX is causal in resistance, then we overexpress LOX and again gave doxorubicin, and whenever we overexpress LOX, doxorubicin become less effective, showing that cells are more resistant. Of course, all these experiments are done in 3D culture in the presence of collagen 1. And collagen 1 is the major substrate of LOX. Again, similar to LOX overexpression, if we put the cells in collagen, cells are more resistant to doxorubicin. And here, if you look at the apoptosis, if the cells are cultured without collagen, they are dying upon doxorubicin, but if you culture them with collagen, they are not dying. So this shows that LOX and also it is substrate collagen confer doxorubicin resistance. And then if we block LOX with this inhibitor, we see synergistic growth inhibition, and also we see reduction in collagen cross-linking and fibronectin assembly as we expect. And importantly, doxorubicin has intrinsic fluorescence, so we can detect the fluorescence of doxorubicin in 3D culture, even in animals. And when we give LOX inhibitor and doxorubicin, what we see is actually whenever LOX is blocked, doxorubicin content or penetration increase. And also as a result, we have substantial reduction of FAK and SARC survival signaling. So these are 3D culture experiments. What about in animals? So what we did here, again, we took our xenograft models. Again, we developed resistance. This time we gave less dose to get the resistance faster. As you see, again, we have response. Then when resistance starting to develop at this dose, we include LOX inhibitor here. And as you can see, we can stabilize the tumor growth compared to the doxorubicin alone group. And this increases the survival. And also, if you look here, whenever we give LOX inhibitor, doxorubicin becomes more effective all over the body, actually, then body weight is decreasing. And also, this is validated with reduced LOX activity. And importantly, whenever we have LOX inhibitor, we see that there is reduced fibrillar collagen, showing that LOX inhibitor is doing its function. Then we wanted to test them in more human setting. And in this case, what we did is we got the PDXs. So from patient tumor, we have the PDXs in mice. Then we generated the organoid culture. We have also gene expression and mutation data of these PDXs. Then before doing the expensive in vivo testing, we tested them in organoid cultures. Here we had 15 of those PDXs, and here LOX expression, here hypoxia. And importantly, again, in the context of precision medicine, we are looking for patients who have high LOX expression and high hypoxia score based on our data. And we had a couple of them actually here. We have chosen one of them, and we tested the combination of doxorubicin with LOX inhibitor. We see that there is a significant reduction of growth in organoids. Then when we move to in vivo with these PDXs, whenever we have combination, we have nice stabilization of the tumor. And again, body weight is decreasing to acceptable range, and also fibrillar collagen reduced. And if you remember this cartoon, we also show that whenever the stiffness increases because of LOX, drug penetrance is reduced. And when you target LOX with BAPAN, what you see is doxorubicin is penetrating much better, and you see that LOX inhibition is actually making doxorubicin more available to the tissue. And also this is accompanied by reduction in phospho-FAK and SARC phosphorylation. And then the question was, if we target FAK or SARC, can we get similar results? Yes, indeed. Both in 3D culture and in vivo, what we see is whenever we combine doxorubicin with SARC inhibitor or doxorubicin with FAK inhibitor, we see substantial reduction of tumors. And since we have been working on non-coding RNAs more than a decade, and we know that pathways are fine-tuned by these microRNAs, we suspected that there could be certain microRNAs regulating these 3 genes at the same time. We identified 8 of them. And one of them was MIR142. Its expression was reduced in our resistant tumors. And also in patients, it is high expression predicting better survival because we expect it to be a tumor suppressor. And its expression is reduced by hypoxia. And it is targeting these 3 genes directly because the 3' UTR assays show that. And in vitro or in vivo, we see that this microRNA, when it is overexpressed, reduce HIF1, LOX, ITJ5, and the downstream significantly. And most importantly, when we overexpress this microRNA and give doxorubicin, we can increase the efficacy of doxorubicin. So in summary, we published these results last year. And what I show you here is upon hypoxia in chemo-resistant tumors, we have induction of HIF1 alpha, then HIF1 induces LOX expression. At this point, LOX can do several things. One of them is go outside the cell, do collagen cross-linking, and then activate integrin signaling and chemoresistance. Then also it can block the drug penetration. This will further lead to no inhibition of FAK-Sarc and chemoresistance. And also, as I show you here, LOX can regulate the expression of these genes. And here also we show that microRNA, which is normally repressed by HIF1 alpha, is not repressed because there is a huge induction repressed. And since it is repressed, HIF1 cannot go down, LOX cannot go down, and ITGF5 cannot go down. This further contributes to chemoresistance. So if you target LOX with BAPEN or FAK-Sarc with these inhibitors, you can overcome resistance. But every study has limitations. So what about other chemotherapy agents? So this was more like doxorubicin. So what is, how is the mechanism of gene regulation by LOX here? So we know more at this point. So hopefully we will publish this soon. And then most importantly, what was specific non-toxic LOX inhibitors? And if you remember this cartoon, our idea was bring these patients here to there. And we know actually from at least 2 different data sets that 50% of the patients expressing high LOX have residual disease. So this means that if we have a specific non-toxic LOX inhibitor, we may bring these patients from here to there. And for this purpose, in the last 2 minutes, I will show you a couple of slides how we identified our novel LOX inhibitors. So we did a screen with more than 5,000 compounds, and we identified 20 hits, reducing the LOX activity without affecting the cell viability because we don't want toxic molecules. And then we tested these molecules in 3D collagen assay that I showed you in the previous parts. And we identified 5 of them leading to doxorubicin sensitization. And these effects could be all indirect, right? So that's why we wanted to test with recombinant LOX protein as well as sister protein LOXL2 to see if there is direct interaction and reduction of enzymatic activity. 2 of these molecules, 32 and 29, reduce LOX significantly without much affecting LOXL2. And our inhibitors are used at 10 micromolar compared to non-inhibitor BAPEN, which is 10 millimolar. So at 1,000 times less concentration, our inhibitors are effective. And also, as you can see here, we can block the collagen cross-linking and fibronectin assembly very efficiently. And then we did structure activity relationship analysis with our collaborator, medicinal chemist collaborator. This was our parental molecule. By changing certain substitutes, we could obtain 3 molecules which have better LOX activity, and they have also better selectivity. And also normal cells are not affected by this new analog. So there was a little effect at 20 with this initial molecule, but not with this one. So then also, if you look at the collagen cross-linking or fibronectin assembly, we have very nice reduction of these. And finally, we did also PK analysis, and we compared our inhibitor with the recently published LOX inhibitor in 2020, and we have much better PK parameters. Our molecule has a very good half-life and very good concentration, plasma concentration, and less clearance. And also with respect to toxicity, from 25 to 200 mg per kg, we don't see any body weight loss. This is now done for 30 days. Also in 30 days, we don't see any toxicity. And if you look at the organs, you see no major effect even at 200 mg per kg. And so we are now here. So we are doing in vivo drug sensitization and metastase experiments using our patient-derived xenografts and transgenic models. So this is our pipeline. So as Aykut Hocam showed, there could be different pipelines, but in this case, we start from screen with a different approach, but again, very similar approaches are done. But there is, of course, dead valley in this process. So currently, we have a molecule which is our candidate for lead optimization. And initial data show that actually it leads to chemosensitization, and it may also work as a single agent in some tumor indications, which I cannot share now, maybe next time. So finally, I would like to thank the people from my lab. especially Özge Saatçi drove this LOX project, and with the help of Umar, Özge Akbulut. And this project will be taken over by Metin. He just joined our lab. I would like to thank to our funding agencies, NIH, American Cancer Society, Meriki, and Halozyme. Also, I would like to thank our collaborators. And also, we have multiple positions open in the lab, student, postdoc, visiting scientists. If anybody interested, please just drop me an email. And thank you for your attention.
Thank you very much, and good luck for your very promising candidate. Are there any questions?
Or you can type. I have a question actually. Have you chance to check ROS levels of these compounds, effect of their effect?
Yeah, this is a very good question. So, as you know, doxorubicin and some other agents are inducing ROS, hydrogen peroxide. And indeed, what we believe is the ROS is a major part of mechanism of how this is all happening. And we have a current grant application that is actually one of the aims of that grant. So we will be studying that in detail soon.
Thanks again.
Thank you very much.
Professor Evren Alıcı, and he's also another good model for our students as well. Since I think when I first met him, he was 4th grade medical student. Am I right, Evren?
Yeah.
And I can easily see that he was really motivated and research-oriented medical student. And at the end, I'm very honored and proud to present him to you. Dr. Alıcı, after graduation from Ege University Medical School, he went
and he completed his PhD at Karolinska Institute, and he worked on a really well-established gene therapy. Lentiviral and retroviral gene transfer, stem cell transplantation, and immunology. In addition to PhD, he also became a surgeon, and he worked in clinic as well. So he's working at Karolinska Institute as a research group leader at Department of Medicine, and also he serves as co-director of NextGen NK, which is an international competence center for the development of next generation NK-based cancer immunotherapies. So he has several publications on this subject, and also he's currently leading a group of more than 20 scientists. And I think I should mention about 2 more things. He's actually in the planning and design of the first clinical study with actually genetically modified cells in Sweden, and also he's responsible for the first in-man autolog NK therapy clinical trial that was classified as advanced therapy medicinal product use. So Evren, stage is yours, and thank you for accepting our invitation.
Thank you, Neşe abla.
Thank you, Neşe abla.
Humbling.
Humbling.
So number one, thank you, Mehmet Hocam. Thank you, Neşe abla, for inviting me. It's really an honor. As you said, we've known each other for quite a while now, and you know how I was. I think it was good that I got into research, because this is really what we were all burning for. I heard this morning when I was out of curiosity, listening to the history of medicine in Izmir, one of my professors was Sezen Koşay, and I didn't know that she passed away. She had a huge impact on me during my medical training, and she actually managed to explain to me the drug-drug interactions, the general concepts between basic pharmacology, and she took the time. She had the patience. And her son is a professor at Dokuzeri, as you may know. He's also a very humble, really hardworking person. I guess some things do go genetic. So with that, again, let me start. I usually start with the acknowledgements because I usually don't make it. But this is the research group. And I'm going to today mainly discuss with you some of the clinical data. I will go the other way around compared to our previous speakers. And I should also mention to Uğur, we are actually testing the TK216 in myeloma. So it's a really interesting drug, but we can maybe discuss that at a later occasion. In collaboration with Oncternal and combination with NK cell-based therapies. So majority of the clinical data I'll show today is in collaboration with Haret, who was my former postdoc, and Hans Gustav, who was my PhD mentor. And this is the group members. Actually, this was pre-COVID. I think the group has grown now. We're probably 25 people, somewhere around. And these are all the members that have had significant, our collaborators, Jeff Meller, Sarah Cooley, now in Fate Therapeutics, Tom Temple, who's phenomenal sarcoma surgeon in Florida, in University of Miami now. And these are the alumni. Some of them are from İzmir, some of them are from Istanbul. So Nesha mentioned a little bit, and as usual, I need to do my disclosures as well. I founded 5 companies. One of them is a vaccine company, a flavivirus vaccine company, basically using the existing vaccines and combining them to increase resistance to flavivirus infections. Hantavirus is a big issue in Sweden, and tick-borne encephalitis as well. So we're really looking into that there with epitope overlap, especially the NS5 protein. And then we have a couple of bispecific engager projects that we actually started with Merck Therapeutics, the German Merck. Now it became its own spin-off, and it's Boston-based. It's called Fuse Bio. And then we have, I founded 2 companies called Ycelix and Ygene Bio. And this is very mobile, but I have 4 or 5 different advisor roles. Some of the companies I funded and left, basically, they're on their own. Some of the companies really brilliant work that I find exciting. Most of them are almost all of them are in lentiviral, retroviral, gene modification, or cell therapy. And this is what Nesha was talking about, the Next-Gen NK Cell Competence Center. So what we did was, a couple of years ago, we really started venturing into collecting all NK cell interested parties, both industrial, healthcare, and the Swedish government, to be able, and the European Union, to be able to start a unique competence center that's only building on the immunotherapy and the subgroup of immunotherapy, NK cell-based immunotherapies in cancer, and how to modulate these NK cells, both in autologous fashion and also in an allogeneic fashion, if we can adapt it. So for those of you who are uninitiated to the NK cell field, let me just remove this. So Eva Klein, early 1970s, we're talking 1972, 1973, before I was born, together with Rolf Kissling, who was her PhD student at that time, hypothesized that there is this subgroup of effector cells in mice that actually can kill tumor cells and virally infected cells without prior sensitization. And when they started looking, they initially hypothesized this was a T-cell subpopulation, and a lot of people, when Rolf published his paper in 1975 and his thesis in 1976, they thought it was an artifact. And what he mentions in the paper as well, in the initial paper, this may basically they did extensive studies to exclude the CD3 positive population, and they did identify the NK11 at that stage as a marker. But the mechanisms of NK cell mediated degranulation activity was not clear. And then comes Rolf's and Eva's joint student, Klaas Scherre, who was working on different mouse models, and he was looking into hybrid resistance systems that was initially suggested by a group in Austin, Texas. And when he started looking into these H2 deficient lymphomas and their engraftment, he sees that there is a missing context. So a lot of people thought there was an activating ligand and an activating receptor, and that was what was triggering. But how do you explain then the absence of something and the lymphocyte being able to attack? So they started hypothesizing. There were 6-7 different hypotheses at that time. But during that period, there was this Russian submarines going into Swedish archipelago. And the Russian submarine silhouettes were published, the Swedish submarine silhouette was published on the newspaper for the fishermen and for the public. If you see the silhouette, it's a Swedish submarine. But if it looks anything else, it's a Russian submarine. You should report it to the Navy. And with that, when Klaas was writing his thesis, he hypothesized actually that there might be, that the cell is normally recognizing something, an inhibitory ligand, that is keeping the cells in check. And this is how the whole missing self hypothesis started, together with Klaas's student at that time, Hans Gustav Ljunggren, who hypothesized that it could be the HL or MHC molecules that might be the self strategy, basically recognition of self. And after that, we identified many years, 25 years later, killer immunoglobulin-like receptors were identified, the marine counterparts Li49s were identified, and NK cells became and since all these people are from Karolinska Institute, from the discovery, NK cells really is a major research area in Sweden. But until my move, there were no clinical trials that were planned. Now we get a much better understanding of NK cells as our flow cytometric phenotyping and proteomic understanding gets better. And this is what Nutsa, one of the PhD students, nicely drew for me. Trying to segregate, it's impossible to do this, but somewhat reductionistically, trying to understand different receptors on NK cells. We have the activating receptor group, very dominant, and usually CD3 zeta downstream pathway, but there are other pathways in NK cells like DAP10 and DAP12. And the NM1, for example, utilizes an activating motif that is completely independent of the others. Then we have antibody-dependent cellular cytotoxicity. This is going to be really important during my call. NK cells are one of the major effector populations when you have an antibody against something. They are very good in exerting antibody-dependent cellular cytotoxicity. So, and this is actually one of the important ways that they control different tumors or viral diseases that you were previously exposed to. There is a death ligand-mediated killing. This is relatively limited with the exclusion of trail. And adhesion receptors that are really important both for extravasation and synapse formation. And then they respond to a lot of different cytokines. So again, Neşe abla just mentioned, I could have stopped actually there. We basically started thinking, okay, there is the missing self. Is there a concept that we can look at the missing self within an autologous setup? Our genotype dictates our certain HLA and our KIR, the immunoglobulin-like receptors that I was mentioning. But these actually do not come up in correlation to each other. KIR acquisition during maturation, lineage commitment, is a stochastic process. So with that, we started thinking, okay, what is the best model? I was working with myeloma patients at that time. So we took myeloma, which has an autologous stem cell transplant component, which I can really look into lymphoid differentiation and lineage commitment. So that's what we did. we took NK cells, we identified the strategy first. I skipped that, but that's a 10-year process and 2 PhD students. We identified a GMP compliant closed system that we can expand NK cells and activate them and recover their phenotype. So they upregulate the receptors that they normally downregulate in the tumor microenvironment. And the whole hypothesis builds on, in the absence of tumor, mimicking an inflammatory milieu renders the daughter cells upregulate the activating receptors again. And when we optimized that and we applied to the clinical trial, we went through the whole process just like a drug development. This is actually considered as a drug in European Union guidelines and in Turkey as well now. So we don't really touch the patient except getting 1 unit of peripheral blood donation. Initially, we wanted to get apheresis, but the patient started complaining that they need to go to the blood center for that. So we said, okay, we won't do that. We will do a standard peripheral blood donation. So all patients that are eligible for an autologous transplant at diagnosis were asked. And except one, all of them accepted. And then we expanded those cells and froze them. While the patient, we wanted to wait for the patients to relapse. Of course, the Swedish Medicinal Products Agency immediately said, you cannot wait that long. You need to have a certain maximum time period in your clinical study protocol. So we said 100 days, which was probably too early, but it was good anyway. And instead of a standard cohort dose escalation scheme where you do dose 1, 3 patients, dose 2, 3 patients, dose 3 patients, what we did was all patients got dose 1, dose 2, dose 3 at weekly intervals. And this actually proved to be really important, which I will show now. And we followed these patients. This was a first-in-human study, and it is the first time that actually the cells were administered in Sweden. So here you can see how these patients do with regards to the M component. As you know, myeloma is a plasma cell disease. What can you measure in a plasma cell disease? The best biomarker is immunoglobulin levels. So we measured that and looked at the infusion times, and you can see that there's a correlation and reduction of immunoglobulins. There are non-secretory groups as well. So we looked at those as well. 2 of the 6 patients were non-secretory in phenotype. And basically, they were non-detectable during the infusion time, but the others gave a response. Except one patient here, I don't know if you see my cursor, but patient 105 actually relapsed after 6 months. All the others stayed in residual state for a relatively long time. Then what we did was we took the VDJ region of these patients and sequenced it and looked into high resolution MRD with next generation sequencing. And looking at bone marrow aspirates 2 weeks before and 4 weeks after. Since it's bone marrow aspirate, you cannot do it at very tight intervals. And you can see that there is also a trend there that these cells are going down in time for even 4 weeks after infusion. But the most important question is, how is the overall survival, progression-free survival? The progression-free survival in these patients was, the treated group was 34 months. When we look at the year match cohort, same group that went through transplant and non-NK cell treatment, that would be around 26 to 28 months. So there is a 6-month betterment. And overall survival was 100% at, I think we have now 7 years follow-up of these patients. And still, the overall survival is 100%. Then we did something maybe quite intriguing and different. So since we are phenotyping these cells extensively, and we're infusing in the non-dominant arm most of the time, we could actually look from the other arm how many cells escape the lung and go into circulation, because that's an open question for all cell therapies. How many of these cells get stuck in the lung? How many of them actually make it into circulation? So that's what we did. We collected blood pre-infusion, 30, 60, and 4 hours after infusion at each infusion. And we could actually follow these cells phenotypically because of their activated status and Ki67 positivity, because we pushed them for proliferation. And here on the right, you can see that the first dose doesn't really make a significant dent, but you can actually see patient-dependent responses that you can detect these cells quite efficiently in circulation in the non-infusion arm. So they do very rapidly go into circulation and pass the lung. Probably most of them are still in the lung by that time. And that's what we did. This is called Tisney analysis. It's basically a random distribution initially, and you can look at different subpopulations. Relatively agnostic strategy to look into different subpopulations, and basically a flow cytometric analysis. And here you can see three different populations that I would like you to focus on. This is infusion 1 group. This is infusion 2 group, infusion 3 group, and infusion 4, follow-up group, long-term follow-up, 2 weeks and 4 weeks long-term follow-up. And here you can see early on, there is nothing, it's slowly coming, there is a population that is slowly coming up, and it's really filling up here. Here, just before the second infusion, we get a significant population. Interestingly, this population is Ki67 dim. So this is really still having the activated NK cells in circulation, but it is cumulatively increasing with the doses, and we can still detect them 4 weeks after the infusion. And these cells really seem to go into the bone marrow and reside there, where we want them. The other thing we did is a detailed serum proteomic analysis of these patients from the same blood collection. So we did an Olink analysis, basically looking at the immuno-oncology panel, relatively agnostic again. And you can see that granzyme B levels really peaked during the first, second, and third infusions. And if you look at the data here, granzyme B levels correlate very nicely with the infusion. So we think this could be a nice predictor of the circulating activated NK cells and also the NK cells in the bone marrow. Most interestingly, and this is what excited us, we compared the diagnosis granzyme B levels in the bone marrow and the bone marrow aspiration 4 weeks after the infusions that 4 patients gave permit to get their bone marrow aspirates. We could see that there is a significant increase in their amount of granzyme B, which suggests that these cells are still around, and phenotypically they are. Now, I mentioned anti-CD38, and it's important because we have a lot of monoclonal therapies. We just listened to initially talk about Herceptin and trastuzumab. Similarly, in myeloma, we have CD38. And it's actually very comparable. CD38 is expressed on a lot of different tissues also. But you can see that this is pretty efficient treatment. And we started this early on. So the first patient we included was 2014 as a hospital exempt. And then we started running this routine. This is one of the first papers, I think, that showed that daratumumab anti-CD38 retreatment works. One of the questions in the field was that the epitope is lost or the CD38 intensity goes down. But it's not the case, actually. When you look at the treatment and relapse, this is the patient's tumor burden measured by M component with different drugs here. And you can see it goes up and down with different treatments. With daratumumab, you can really keep it low. And once you see the patients relapsing, increasing the dose actually helped. And the CD38 expression is still there. One of the things then we started asking, what is actually happening? Why don't these patients respond? Why do they need a treatment interruption? And the answer, in our opinion, is this. It's the decrease of NK cells, the ADCC competent effector population that is getting lost immediately after the daratumumab infusion. And although they're coming continuously, the CD16 is cleaved, and the NK cell counts are kept low, although there is some in the bone marrow. So what does this mean? This means the following. We have a lot of patients that here you can see the tumor reduction that respond really well, and they don't usually get side effects. Then you have the medium population that responds somewhat, gets a tumor reduction, and gets some of the side effects. What you lose in NK cells is really the infectious complications, varicella zoster, cytomegalovirus, bacterial infections like E. coli. And the ones that are really suffering are the ones that are progressing or stable disease. So once we realized these patients are not doing fantastic with this, we started thinking, okay, why don't we support this with an NK cell product? And we approached Sanofi Genzyme at that time, which had a CD38 antibody, and we designed a randomized clinical trial where we give antibody in one arm and antibody and ex vivo expand NK cells in the other arm. So I'm excited to say this trial is ongoing now. We actually included our fourth patient. It's going to be 60 patients in total, 30 patients in each arm. In Sweden, that's a big size for a clinical trial compared to US. And I think it's going to finish by the end of next year if the inclusion goes this way. So I'm going to probably skip this, but just to give you some sense of where we are going, is we worked a lot on lentiviral gene delivery to NK cells, and NK cells are innately and inherently resistant to lentiviral gene modification because they are evolved to kill RNA virus infected cells. And one of the reasons is they have very high TBK1 IKK epsilon. And once we identified that, we screened for small molecules. We identified a small molecule that increases the transduction rate at a very low MOI, even at resting state. So very low number of viral particles can lead to a good infection. And this actually enables clinical development of gene-modified NK cells. T cells are much easier to gene-modified compared to NK cells. Funnily enough, this is a serendipitous finding. It also helps the same small molecule. It also helps the re-guide detection of the guide RNAs, both with the Cas13 systems and the Cas9 CRISPR-Cas9 based systems. In this setup, we are knocking out beta-2M, which is the most established guide in our lab. And you can see that even primary hepatocytes that are notoriously famous for spontaneous knockouts, this is without any selection, like this is spontaneous knockout. And you can see that we can actually get pretty feasible knockouts up to 30% with the VIOS. And this is tumor infiltrating lymphocytes, also very, very helpful because you have very few of them. The other thing that it enabled was that we could, unlike T cells, we can clone actually all NK cell receptors that I was showing you in the beginning of the presentation. So that's what we did. Tolga, one of the PhD students at that time, took the challenge and really synthesized all these and sent it for synthesis. We got the vectors, we started testing them. Tolga went with the NK92 strategy, we went with the primary NK strategy, and started screening for their tumor responsiveness. And this is really interesting. So this is really myeloma patient group, 1 to 6 patients here. And all the same patients that we followed at diagnosis, relapse at transplant, first relapse and second relapse. So you can see, for example, when we upregulate some of these, you really get functionally responsive NK cells. The redder, I hope there is no color blind among you, but the redder is higher degranulation. The greener is less degranulation. Spontaneous is baseline, basically. And you can see that NKG2D upregulation in every line of treatment is very helpful. But interestingly, the NM1 is the only receptor that is downregulated, and when upregulated, confers an anti-tumor activity during peri-transplant period, which is really, really curious. The other thing we started looking into, I wish we covered this, we thought about this earlier, is small non-coding RNAs, basically double-strand RNAs that can trigger RIG-I. As Amaja mentioned this morning, a little bit about the RIG-I and Toll-like receptor pathways, but we really focus on the RIG-I MDA5 pathway. And this is one of the positive controls. It's also used in VLPs. It's also used in other strategies. We use it here for boosting immunotherapy. And it does, very interestingly, direct delivery of this to effector cells increase perforin and granzyme B, which is the cytolytic molecules in these effector cells. And that confers a good cytotoxicity after a long time. With that, I want to finish by one of the final projects, the iPSC project that Neşe abla mentioned. This is what the granzyme B upregulation leads to, basically. It's a lower conjugation time. So these cells seem to kill faster. And the reason they're killing faster before they become anergic is they can attach and detach faster. And that suggests that granzyme B has actually a role in the CSMAC, the central immunological synapse detachment, which makes sense in retrospect. We also tested in animal models, which I won't get into. And finally, we are reaching clinically relevant effector doses. With that, this is what the field is going towards. All the iPSC NK field is going towards different introductions of class 1 knockouts. Of course, when you knock out class 1, as I was saying in the beginning, you get NK cells rejecting the graft. So that's very complicated. You can also you probably need to knock out class 2 to avoid CD4 responses. And then you can put inhibitory ligands to avoid NK cell recognition and killing. HLA-E is the most famous one, but you can use PD-L1, CD47, etc. The problem is when you use these in an effector cell, it also through cis interactions affect the effector cell's activity. So it actually, the graft becomes useless for us. In a liver transplant setting, fine, but in an NK cell transplant or a T cell transplant setting, it doesn't work. So that's what, then we went into a systematic screening of all the ligands we can think of, how viral evolution has thought about it. We took galactins, UL-18s, different tegument proteins, ran cytotoxicity assays, and identified one that basically completely abrogated NK cell mediated killing in the tumor cells. You can see a 10-to-1 effector target ratio, even we don't get any killing. And T cell responses are the same, no cytotoxicity whatsoever, even at a 100-to-1 ratio after an MLR assay. And when we engrafted this into mice, we could see actually they can evade immune rejection very efficiently. This is a PBMC ADCC model. On the right is the control setup, which we can keep the cells under MRD. On the left, you can see that they escape the immunotherapy very efficiently. And it doesn't affect the effector cells negatively. It's not an inhibitory ligand. So once we identified this synthetic ligand, one of the things that we always think about is, can we generate new CARs that can kill, but that doesn't cause fratricide? Imagine that you have an effector cell that expresses the ligand, but it doesn't actually through fratricide kill each other. It only is targeted towards the tumor. And this is where I'm going to stop. I think this is where we are really happy about. We have the clinical CAR38 product with the UC, and this is really what we are pushing now with the iPSC-derived cells. I would be really excited if there is any interest as a postdoc. We have now saturated our PhD quota. I'll stop here. We have saturated our PhD quota, but I'm happy to take any applicants if there are.
Thank you. For this inspiring talk. Is there any question from audience? Or online? Yeah.
I lost you for a second.
Okay. No question.
Okay, everything was
really clear, as far as I understand from audience. Thank you very much. And
thank you.
Good luck.
Thanks.
It's really exciting, and we are looking forward to hear results soon.
I look forward to that too. Positive or negative, let's get them. Thank you.
Thank you.
Last speaker of the session and also symposium is Professor Fatih Mehmet Uçkun. I'm very glad to present him to you. First of all, I'd like to thank to him for accepting our invitation.
Let me introduce you briefly, and then we can listen your talk. We will be really happy to hear your ongoing research. After completing his medical education at the Heidelberg University, he moved to United States to continue his training as a postdoc, and he worked in immunology and bone marrow transplantation at the University of Minnesota. And I think at the same time, you continue to your residency training in pediatric health and diseases, and you become a pediatric oncologist, as far as I understand from your CV. And then you also continue your education as a professor at the Faculty of Medicine at the University of Minnesota, and also a chair of honor was established for you, and you become a chairman of that department. Its name is Minnesota Biotherapy Institute, and Professor Uchkun worked between 1995 to 1997 to that department, and then he continued his education, not education actually, he continued to train young scientists as a professor in Minnesota, and then he became a professor at the University of Southern California between 2009 and 2015. And also he continued his scientific studies as the head of the Leukemia Lymphoma Research Department in the Pediatric Cancer Center of the same university. Between 2008 and 2016, he also took part in many leadership positions in the nanotechnology program of US National Cancer Institute. He has several patents and more than 500 publications, and also he has really prestigious awards. And I'd like to mention one more thing about him. He also trained several Turkish students and also specialist residents in his laboratory, and this is really important for us. Thanks also for your contribution to young scientists and for your support. Your CV is really impressive, and I have limited time, so I'd like to give the stage to you. Thanks for your presence to this meeting.
Okay. İzmir'i ziyaret edebildiğimizde COVID-19 sonrası sizlerle şahsen de tanışmak isterim. Öğrencilerinizle de tabii ki şahsen tanışmak isterim. Going back to my presentation, I will try to share my screen, and I hope it works. So let me see if this, please let me know if this is working. Here we go. Can you see my screen?
Yes, we can.
Okay. Okay, then I will. Okay. The title of my talk today is Clinical Impact Potential of CD3 Engaging Bispecific Antibodies as Biotherapeutic Agents for Hematologic Malignancies. First, a few financial disclosures. In this area, I had some out-licensed patents that creates a conflict of interest that I need to disclose. And I have some affiliations with private entities. And I will, of course, attempt to make sure that the presentation is balanced and is not affected by these affiliations.
Also, some of the statements, while I'll try to avoid them, may be considered forward-looking statements in the US, this private securities litigation reform. So I will try to keep them to a minimum. With that intro, I would like to start talking about acute lymphoblastic leukemia. This is, of course, as you know, the most common form of childhood cancer. And most of the patients with ALL have the so-called D lineage. That is because the malignant cell population in ALL represents a neoplastic expansion and malignant expansion of B cell precursors that are frozen at the discrete stage of B cell development. So these are cells that did not differentiate further into mature B cells or plasma cells. They became growing B cell precursors with or without some genetic abnormalities and cause leukemia. This slide shows you what in the United States and now also in Europe is being referred to as the pediatric paradigm. When I joined University of Minnesota, the slides were being presented to me. We were at that time below the 80% level in 1984. We were talking about the time before Nesbitt and after Nesbitt. A group of physicians around pediatric oncologists, including Mark Nesbitt, made a huge difference from 1970s going to 1980s. If you can see the survival rates, there has been a huge jump. And this success has been the result of major collaborative effort among pediatric oncologists nationwide and globally later on, including many other countries. And the challenge has been, just imagine, the challenge has been that there were no drugs, right? So the reason for the success has been trying by trial and error 12 different drugs that were not designed for pediatric oncology or pediatric leukemias, and trying them in certain combinations to make them work. We need to remember it. If you look at the success rate now, it is now above 90%, and most of these patients are considered cured. So there's a huge success going from less than 10% survival in 1970s to all the way now above 90% cure rate among these children. Now, are there challenges? There are still some challenges, and some of these challenges relate to the side effects of the intensive therapy that's given to the children, and that's one challenge. The second, of course, major challenge is relapse. The second most common cancer happens to be relapsed ALL, so patients can still have a recurrence. And when they have recurrence, to put them back into remission, to salvage them, to achieve long-term survival is very difficult. It comes also at a major cost for these children due to the side effects, development, both mental development and physical development, endocrine problems, etc. So there is still a major need, despite this wonderful advance in treatment of children with ALL. There's still a need for new treatment modalities. Now, while the results have been so promising in terms of survival for children, the same has not been true for adults with ALL. Indeed, that's how I got into the field of ALL, was by losing someone very special to young adult ALL at that time. So if you look at I don't know if you see my arrow, but if you look at the complete remission rates showing the success of the anti-leukemic activity of the chemotherapy drugs used, a good percentage of patients achieve complete remission. So that part is not a problem, not a limitation. But when you then look on the right side at the survival rates, there are nowhere where the pediatric survival rates were. So there was a big difference in the outcome, in the survival outcome between children and adults. And people were trying to understand what can be done about it. And one of the approaches was to consider having pediatric protocols, pediatric treatment regimens used for adults, with the thought that maybe if pediatric oncologists treat adult ALL patients, maybe perhaps the results might be better. But what we then have realized is what I'm going to show you in the next couple of slides. Just one point, when these patients relapse, even the not very high poor prognosis population, Ph negative population, their outcome is dismal. So once an adult ALL patient relapses, the outcome is less than 10% long term. And this result is also very important to remember. The question is, why is that? And it turns out that the favorable subsets of ALL are more common among pediatric patients, and the unfavorable subsets, types of leukemia, such as Philadelphia chromosome positive, BCR-ABL positive leukemias, and Ph-like leukemia are more common in adults, 5 times more common in adults than they are in children, if you look at the Ph positive ALL. So that means then the challenging leukemias are more common in adults. Adults, of course, especially in older age, they are going to have more comorbidities. They don't tolerate some of the best drugs available for ALL, such as L-asparagine or derivatives thereof. So it was a major challenge. Therefore, the paradigm for treating adult ALL has shifted from chemotherapy to immunotherapy. And this was spearheaded by Dieter Hölzer's team in Germany and his group, as well as Hagop Kantarjan at MD Anderson. And the idea here was, can we, rather than trying to give so much chemotherapy that apparently does not offer us long-term survival at the same level long-term survival is achieved in pediatric patient populations, for example, with augmented BFM type chemotherapy, can we do that by focusing more on antibodies? And there have been several antibodies targeting antigens on bilineage leukemia cells. Most of our work was focused on CD19 and antibodies that I developed when I was working under Muzaffer Aksoy at Chapa University. And CD22 and CD20, they are also applicable for ALL, but not as attractive as CD19, which is expressed on more than 99% of pediatric and adult ALL cases. And the question is, what can you do with these antibodies? In this cartoon slide, I would like just to mention that these antigens are expressed on the surface. They're expressed also inside the cells, but the relevant part is that they're expressed on the surface of these leukemia cells. And you can attack them with either unconjugated antibodies. Of course, you know that from lymphoma therapies, Rituxan used to attack CD20 antigen on the surface of leukemia lymphoma cells. You can use an immunotoxin. You can use an antibody that's linked to a toxin. You can also use immunoconjugates, antibody drug conjugates. So there are many ways to target the surface of leukemia cells to kill them. More recently, there has been interest in immuno-oncology, and there's been interest in bispecific antibodies. These bispecific antibodies are a little bit different. They don't typically activate T cells. The T cells in our immune system are activated only if they are redirected to cancer cells. So the antibody here on this slide, on the right side, you can see, if you look at the T cells, they have CD3 on their surface. The bispecific antibody, in this case, blinatumab, CD19, CD3 bispecific antibody from Amgen, binds to CD3 positive T cells and licenses them. It redirects them, brings them to the close vicinity of the target leukemia cells, in this case, target leukemia cells that express CD19 antigen. And this interaction, this close interaction, then enhances the likelihood for the T cell to destroy the cancer cell that is being attacked. So in this cartoon slide published by Bassan in Blood back in 2012, you can see the mechanism of action. So you have the T cell with the CD3 antigen on it. And then to that T cell then bound is the blinatumab, the bispecific antibody. So now you have these T cells carrying the antibody. The antibody has not only the CD3 portion, it also has a CD19 portion. So CD19 portion binds to leukemia cells and creates this cytolytic synapse, which allows in this very close vicinity then T cells to kill leukemia cells. Leukemic cells die, and you get a cytoreduction and potentially remission in these patients. Now, the first clinical proof of concept that bispecific antibodies can actually work. The idea was good, but will it actually work? Can they work as good as chemotherapy? This was tried in adults with B lineage ALL, and the bispecific antibody performed very well. The results were published by the MD Anderson team in New England Journal of Medicine back in 2017, and it really changed the adult paradigm. So we were talking about pediatric paradigm, intensive therapy, with a combination of drugs based on trial and error, whereas the adult paradigm shifted from chemotherapy that wasn't working to focusing on leveraging what antibodies can offer. In this case, blinatumomab was the antibody, the bispecific antibody tried. So in this study, blinatumomab compared to standard of care chemotherapy. Different chemotherapy choices, investigator's choice. Whatever the site considered standard of care was used. Identical treatments. So you have either induction with antibody or induction with chemotherapy, consolidation with antibody or consolidation with chemotherapy, and then comes remission. After remission, 12 months of maintenance therapy. And this approach actually worked well. And if you look on the right side, just at the complete remission rate, and this complete remission rate includes patients who have a real true complete remission with full recovery of their blood-forming elements, hematopoietic cells, white blood count, platelets, they become transfusion independent, as well as patients who have no more leukemia cells detectable, but they have in their bone, they don't have full recovery of their hematologic cells and platelets. So when you combine all of the complete remission classes, a higher percentage of patients who received blinatumomab, 271 patients were treated, 45% achieved an objective response, achieved a complete remission. And that was very impressive. It was the first time that we had learned, indeed, that bispecific antibodies can be used for treatment of a leukemia patient at very high risk for relapse. And this translated into also survival advantage. So at that point in time, since we did not know what the long-term survival was going to be, once patients achieved the remission, most of them were transferred to a transplant unit to undergo transplantation. So if you look at the survival data up to the time point of transplant, even then, if you look at the 50% survival, it takes much longer for patients who receive blinatumomab to fail, half of them to fail. So the survival difference between the two groups was also was also very clear. So blinatumomab not only induced a higher rate of complete remissions, it also was effective in improving survival. Then the focus shifted in the field to minimal disease. There were two reasons for that. The more leukemia cells a patient had, the more activation of T cells people observed with use of bispecific antibodies. And one of the main side effects with blinatumomab and similar antibodies has been cytokine release syndrome. Also, of course, a side effect seen for CAR T cells, we know from the treatment of COVID-19 patients, very similar to the survival threatening side effects of COVID-19 with COVID-19 pneumonia. So therefore, if you treat patients when they have minimal disease, if they have MRD or minimal residual leukemia, there's not going to be much activation of T cells to cause cytokine release syndrome. And because you will have more bispecific antibody molecules per leukemic cell, you will be more likely to achieve greater success, greater destruction percentage-wise. And therefore, you will have better results, more optimal results with less side effects. So therefore, the emphasis shifted to MRD, but this slide is to tell you that there's also a clinical reason. If you look at the MRD positive patients in remission and MRD negative patients in remission, you realize that it is the MRD positive population that then fails and their survival is going to be worse. And therefore, the thought was to not only then reduce the side effects and increase the effectiveness at the single cell level, but also to try to shift this MRD positive patient population into an MRD negative patient population. And this was tested, and it worked. It was published, and patients who have been in remission received blinatumomab, and after blinatumomab, they actually turned MRD negative. A majority of them turned MRD negative, and this was very exciting. So we then learned that you can actually replace chemotherapy with blinatumomab without losing much, and you can also then treat complete remission patients who have MRD, who have still evidence of resistant residual leukemia that you could not eliminate with chemotherapy. You could then eliminate them with blinatumomab. So this really changed the paradigm in adult ALL. This is just to tell you, after just one cycle of blinatumomab, a significant portion of the patients then became MRD negative. Very impressive results, and really provided the foundation for ongoing research for ALL as well as AML. So the paradigm that I'm talking about then is away from a lot of chemotherapy to a treatment strategy in which you try to give your total therapy in less than 1 year. You give your chemotherapy within 6 months. You try to give them within 6 months. Originally, we were thinking about 8 months of therapy. And the therapies don't need to be as intensive as we believed we had to. And following this chemotherapy, you come in and use a drug, in this case, a bispecific antibody, which we know is as effective as these chemotherapies, but then kills the cells that are resistant to chemotherapies. We know that from the minimal disease studies. And then you give chemotherapy again, just in case some cells did not get killed by the bispecific antibody. So you assume the best case scenario, you assume the worst case scenario, you give some more chemotherapy, followed by, again, consolidation with the bispecific antibody. At the end, then, you can use CAR T cells, for example, or you can use CAR NK cells, or you can use gamma delta cells. You can use cell types that will eliminate cells that cannot be reached by antibody or chemotherapy. So with that introduction, I would like to go into a little bit about into the treatments for bispecific for AML patients. And very briefly, the results have been just published a couple of weeks ago, so I'm going to just focus on the CD123. This antigen is expressed on AML cells as well as ALL cells and many different types of leukemias. We are exploring an antibody-like structure. This is encoded by a single gene. It's a bivalent, so it's very strong binder, much higher affinity to the target leukemia antigen. And it recruits T cells like blinatumomab does. CD3 positive T cells are recruited. And this molecule was tested in a phase 1b study in 46 patients. We just published the results. So I'm going to only discuss some of the main findings in that study, what we learned and where we are heading. So the phase 1 study included a number of multiple relapse AML patients. And in these AML patients, the take-home message was, with blinatumomab, we were able to achieve complete responses, complete remissions, with full recovery of normal cells, which was a good proof of concept finding for this particular molecule. Furthermore, in myelodysplastic syndrome patients, we also achieved marrow complete responses. And the treatment responses to this particular bispecific antibody was associated with a survival advantage, as I will show. Most importantly, as AML is a disease of elderly in the resistant population, especially in the resistant population, it was very well tolerated in older patient population, older than 65 years of age. And there was not much cytokine release syndrome that was difficult to manage. And these findings, of course, provided the basis for the ongoing second portion of the study I will briefly discuss. So in that second portion, which is now activated, we are in the process of enrolling patients. There will be 90 patients being enrolled into 5 different cohorts. In the big picture, we're doing two different things. One is we give chemotherapy first, and then cells are reduced, leukemia cells then are reduced. So once patient is post-chemotherapy, we give the antibody for consolidation. And in other cohorts, we are combining the antibody with chemotherapy in a novel triple drug combination, for example. And finally, we are using it for treatment of MRD, very much like what we did for belantamab. So patients come in with MRD positive complete remission. They are giving the antibody to achieve a complete remission that is a better quality and much less disease. This swimmer plot is to show you the duration of responses in this very small phase 1b clinical proof of concept study. Around a month after entry into the study, patients' leukemia stabilizes. Around the same time, a little bit later maybe, they achieve a partial remission. And then in those patients who achieve a complete remission, then achieve a complete remission with recovery of normal elements, but then they relapse after a while and with small numbers of leukemia cells, and their survival continues. And if you look at the consequences compared to patients who don't respond, patients who are responding have a better survival advantage. So we are trying to enrich then this patient population based on their biomarkers in the current ongoing second part of the study. And as I mentioned, we are using either a novel combination, such as venetoclax plus azacitidine in combination with the antibody, that is one of the cohorts, or we are using, as I mentioned, first chemotherapy induction consolidation, and then we follow that with antibody for consolidation to get rid of the cells, leukemia cells that we could not kill with chemotherapy alone. And again, very intuitive, normal business, taking patients who have MRD positive complete remission, we give them either single agent, the antibody, but in this case, we are giving them oral azacitidine, since this is in the United States, the standard of care, known as Onoreg oral azacitidine, so two-drug combination. And this study is ongoing. We hope to have some results by beginning of next year, and for at least some of the course to see if the results are positive enough to justify a registration study in a randomized setting. And very briefly, something that is of interest to us is to target the tumor microenvironment. In multiple myeloma, an area of interest for me, as well as in myelodysplastic syndrome, the tumor microenvironment includes multiple elements that are immunosuppressive. And one of the elements is myeloid-derived suppressor cells. And these myeloid-derived suppressor cells then suppress cytotoxic T cells. They suppress other elements such as natural killer cells. And they stimulate a negative suppressive T regulatory cell population. And interestingly, this myeloid-derived suppressor cell population has on the surface a CD123 antigen. And so if you look here, then the idea would be you're trying to kill the neoplastic cells, the AML precursors on myelodysplastic cells by targeting CD123. The antibody we just discussed would then hook onto leukemic cells or pre-leukemic cells and kill them by recruiting T cells, and the antibody is shown here in the middle between the myelodysplastic cell and T cell. On the other hand, you can also then target with the same antibody myelodysplastic syndrome microenvironment by attacking the myeloid-derived suppressor cells. They also have CD123 on the surface. This paper is coming out in the next few weeks, week or two, in Frontiers in Oncology. And multiple myeloma paper is currently undergoing the review. So the concept of targeting tumor microenvironment is something of interest in the bispecific antibody field. Finally, this is my last slide. We have advanced now one additional molecule into a clinical stage with a new IND filing and clinical protocol being finalized. And this antigen is of interest to us. Dr. Küçük, Ömer Küçük from Emory University has kindly agreed to be the principal investigator in the study. In the clinical study, the 5T4 antigen, of course, is expressed on many different forms of cancers, including urogenital cancers, lung cancer, colorectal cancer, etc. But it's also expressed at very high levels in relapsed ALL patients, especially pediatric ALL patients express 5D4 antigen on the surface. So, bispecific antibodies can be developed for the purpose of redirecting our immune cells to kill cancer cells. This can be done both for solid tumors as well as for hematologic malignancies. And I will stop with this slide and take any questions you may have. And thank you again for the kind invitation. And it has been a pleasure.
I think Mehmet Hoca has one. And also, I think you can see the chat box.
Hocam duyamıyorum.
Mikrofon kapıda arkadaşlar.
Can you hear me now?
Yes, I can hear you.
Yeah, well, thank you very much. This was a great talk, and it's timely for me because I am growing interest in this chimeric proteins serving many different services. I was very surprised that actually this bispecific antibody is very effective against residual disease. So it came to my mind that maybe a similar approach could be applied for therapeutic or prophylactic applications in solid tumors after the removal of the major tumor. So that it looks like you don't need to have too many targets in order. It's better to have a lower amount of targets in order these type of molecules to be effective. So, are you aware of any activity that tries to use this? It was Binatumoab actually, I guess, in the treatment of solid tumors. Of course, in that case, you would need to change the targeting part of the bispecific antibody. Is there any work in this direction for solid tumors?
Yes. It is a very active area, and many pharmaceutical companies actually spend a lot of money. And one of my students, Hong Mao, is currently in Merck, spending full time together with another student. Their whole project is on solid tumors and bispecific antibodies for the reasons you pointed out. For bispecific, for the solid tumor area, bispecific antibodies that we are currently developing is one of them is this 5D4 antigen directed, and we will be looking at prostate cancer and kidney cancer, so urogenital cancer. So we are starting the trial hopefully within the next 6 months. So when I say within the next 6 months, it typically happens within the next 3 months, but it depends on regulatory agencies. So it is likely to have some challenges. And let me just briefly tell you what those challenges are and how we are trying to address them. When you use these antibodies for liquid tumors or hematologic malignancies, you don't have a problem because you don't have something that is preventing the entry of the antibody into the tumor. When you look at cancers like pancreas cancer, let's say, you have very hard fibrous tissue. You have a wall that prevents entry of biologics, large molecules, into the tumor. So furthermore, there's pressure inside the solid tumors. That pressure then constricts the blood vessels. So the delivery of the antibodies into the inside of the tumor is also difficult. So ultimately, single agent alone, I don't believe will work for solid tumors. I think what probably then needs to be done is you need to soften the tumor. It could be radiation, you need to disrupt the blood vessels. It could be by biologic agents or radiation chemotherapy. It has to be a combination approach, but for the very same reason that you pointed out, to kill the cells that are resistant. Now, going to prophylactic or prevention, you're absolutely right. Around the time of the surgery, right? So if you, let's say, have surgery, you do nanotechnology-based surgery, you make sure that the margins are clear, and now you are giving some antibody to catch the cells that are going to be killed so that they don't metastasize, they don't settle down. Absolutely right. And actually, one of the projects that we are going to initiate is using the antibody together with tumor-infiltrating lymphocytes. So if in those patients that you're going to operate on, you have some cells that you are able to isolate, and you grow those cells, and now you give those cells, and you weaponize the cells even more by using bispecific antibody. So either in combination with cell therapy or alone, it is absolutely very promising. I believe it's just like from a conceptual standpoint, it will be an attractive idea to pursue it. So to the extent that there's interest for collaboration, we will be interested in discussing it further with you. But it is an area of great interest. There's a review article which I'm in the process of putting together. There are hundreds of molecules, so one has to prioritize. And I will be happy to share that with you. It kind of tells you what everybody else is doing and where these studies are with their NCT numbers, the registration numbers for each clinical study. But thank you for the question and allowing me to mention the potential of these in the solid tumor area. But some challenges will remain if you try to treat, not prophylactically, as you said, but if you try to treat a disease that is not removed surgically, it's going to be challenging because of the tumor pressure as well as the capsule around the tumor. Does it answer your question?
Definitely, thank you very much, and I will contact you after this meeting because I have several idea about it, and I need a view about it.
Şereflisiniz hocam. Sağ olun.
Thank you. Also, you can see Professor Alıcı has a question as far as I can see. Can you see his question? Or?
Not yet.
Actually, he says that that APVO436 is extremely interesting. Do you know if the CD123 binding needs to be membrane proximal? Does it block IL-3 signaling?
No, CD123 does not need to be proximal. Most of this antigen, actually, when you look with confocal microscopy, most of the antigen is perinuclear. So it's very similar if you are familiar with CD22, the CD22 story. So there is some expression on the surface, and the epitope we are targeting is on the peripheral part, on the distal part of the antigen. So it's not close to the membrane. On the CD3 portion, you want to have a proximal because of the signaling aspects. But for the targets, typically we try to have a distal portion of the tumor targeted to not have difficulty in a crowded cell cluster. For the antibody to bind. We don't want anything preventing the binding, and we also don't want cis binding in any way or cis-like binding. So therefore, we are not, the CD123 binding is distal, if that is what you were asking.
He says. He thanks and he says, excellent, thank you. So I think you answered his question perfectly. Thanks again for your contribution. And I'd like to actually say a few things about as closing remarks. As far as I learned from the organizers.
1,000 people was actually were applied to join to that meeting, and although I did not get recent reports, more than 10 different areas were established as alive actually contribution of several participants. So I think this meeting was really helpful for several young investigators as well as several clinicians in different parts of Tinaztepe University hospitals were also joined. Another thing, actually our medical students were followed the meeting from their class. This is really good. They were they had chance to meet with all of our guest speakers who were experts on their area, and I believe they are really great role models for our students as well. So also I'd like to thank all of you for being here, and it's really a great pleasure for us to share opening of Galen Research Center with a symposium. Actually, we had really well-known scientists, and it was, we hope to host them as a person in our next Galen Day. I believe we would have chance to organize that kind of meeting with their personal participation. And also I'd like to thank organizing committee, and also there are a lot of people worked to organize this day, and I'd like to thank all of them on behalf of organization committee. And also we had some companies that support our meeting, and I'd like to thank them as well. Mehmet Hocam, would you like to say anything? Add anything? No? Okay. Thanks for your participation. Anything else? Would you like to add?